Right, my very dear students, today we are going to talk about a very important aspect of endocrine system, that is about the thyroid system, right? Thyroid hormone and thyroid gland. Thyroid hormones. Now, thyroid hormone, they are anabolic hormones or catabolic hormones? Yes, doctor. The anabolic hormone, they are catabolic hormone dominantly when they are present in the body in normal level. Yeah, they are anabolic, right? Normally, but of course, when this excess of thyroid hormone is present, it may lead to excess catabolism. Anyway, first of all, we must know what are the major functions of, what is the most important thing about the thyroid hormone is the thyroid hormones are concerned with mental development, mental growth, maturation, physical growth and maturation, basal metabolic rate, and they also maintain sensitivity to sensitivity to adrenergic system, adrenergic system, right? We'll go into detail of all these things, that how it lead to mental maturation, how it lead to physical maturation, how it is maintaining our basal metabolic rate, how it is keeping our tissue appropriately sensitive to adrenergic drive. These things we'll be discussing later. First of all, we'll talk about the structure of the gland very briefly, right? And follicles related in that, right? Now, you know that basically thyroid hormones are produced by the thyroid gland, and thyroid gland is present in the neck in its interior part, just in front of the, just below the larynx, in front of the trachea, right? And thyroid gland is normally a bilobed structure, right? And in this bilobed structure there are two upper pole and two lower pole, and there's a middle isthmus binding the both areas. Like other tissues in our body, thyroid gland has a lot of stroma, right? And its functional tissue, which is a truly functional unit of this gland, that is called follicle. Let me tell you what are the follicles and how do they work, right? Let's look very briefly, very briefly at the structure of thyroid gland. Basically, thyroid gland has these very specialized, you can say, functional unit, which are made of cuboidal cells, and these functional units are basically made of epithelial cells. These epithelial cells make a spherical, you can say, structure which is called follicle. The structure is called follicle, right? And there's too many follicles. There are many follicles in one person's thyroid gland. So now we can say what are follicles. Follicles are specialized structures, right? These are made of thyrocytes, so thyroid functional glands, and these follicles are spherical structures. In cut section, they look like circle of epithelial, you can say epithelial cells, usually cuboidal cells, and these follicles are having in between stroma, especially let's suppose this is the blood supply, capillary network of thyroid structure. Here is also another capillary. And another important thing which is there, that in between the capillaries and follicles, in some areas you find a very unique type of cluster of cells, and these blue cells are called parafollicular cells, because these blue cells are on the side of the follicles. So these blue cells are called parafollicular cells. These parafollicular cells are responsible to produce calcitonin. Calcitonin, a calcium lowering hormone. And follicles, when they are functional, they are responsible to produce T3 and T4, which are thyroid hormones. So we can say from the thyroid gland, there are two types of hormones coming out, T3 and T4, which are derived from the follicles, and calcitonin, which is derived from the perifollicular cells. Is that right? Now, follicles are basically responsible for T3 T4 secretion, and again I'm repeating, parafollicular cells are also called C cells, C for calcitonin. They are responsible for calcitonin handling. T3 T4, they act on every cell in the body. T3 T4, they act on every cell in the body because they're responsible to maintain the metabolism throughout the body. And T3 T4 especially are important for us to mental development and maturation, as well as they are especially important for physical growth as well, right? Moreover, T3 T4, they also keep the tissues sensitive to adrenergic drive, right? Opposite to that, when we talk about calcitonin, calcitonin is basically a calcium lowering hormone. In today's session, we'll be mainly concentrating on thyroid hormones, that is about T3 and T4, right? We'll talk about first of all that what is the basic structure of a thyrocyte, right? Thyrocyte means a single cell which is contributing to a thyroid follicle, and how a particular thyrocyte is collecting the raw material and synthesizing, yes please, synthesizing thyroid hormones, how it stores the hormones in colloid, and eventually how from colloid T3 T4 are absorbed by these cells and secreted into blood, right? So let's go into detail of one follicle, that one single follicle, how does it work, and what is the particular role of a one single thyroid cell or thyrocyte. First of all, we'll talk about how T3 T4 is synthesized and released. Let's suppose I draw one follicle over here, right? As I told you, one thyroid gland has many follicles. Follicles are the basic functional units of thyroid gland concerned with the synthesis of T3 T4. Now, this is a follicle of a normal thyroid gland. As you know, this follicle is made of what type of cells? Cuboidal cells having a nucleus in the center. Now, you know that capillary system is an outer aspect of this, right? Blood flow is around the follicle, right? And this membrane of follicle, this membrane of a follicular cell, right? This membrane is called basal membrane, and this membrane is called, yes please, lateral membrane. Here is also a lateral membrane, and at this point, this part of the follicular cell membrane is called apical membrane, or this is also called luminal membrane because it is facing to the lumen of follicle. The center of the follicle, this area is basically lumen of the follicle. Again, let me repeat it. If I draw this single cell, another cell, right, with different colors, now this part is which one? This is facing on the lumen, right? Here it is, yes. Which part of the cell? Lateral. And what is this part of the membrane? Basal. Is that right? Now we have to see what are the basic principles involved in synthesis of synthesis, storage and release of thyroid hormone. Step number one is that these cells have to take the raw material from blood side. It means it's the basal side of the, yes, it is the basal membrane which is collecting the raw material from the capillary side or interstitial fluid. So step number one should be in synthesis that it should collect the raw material. Then step number two should be that within the follicular cell, within the follicular cell, there should be synthesis of thyroglobulin, right? Raw material like iodide we have taken from the blood. Then we synthesize the thyroglobulin. Then step number three is release of thyroglobin into where? In the lumen. There certain important synthetic steps occur which will go into detail. Then there is a storage phase, that thyroglobin and its associated T3 T4 are stored within this luminal area, and this luminal area, when it is full of thyroglobin, that thyroglobin is called colloid. That is called colloid. It means this is the colloid which is secreted by every cell, and this colloid is what? Colloid is basically, yeah, what is this colloid? Thyroglobin, right? And as a part of thyroglobin, there is synthesis of T3 T4. Then again, listen. Step number one: take the raw material from the interstitial area into the cell, and this function is present into which part of the membrane? Basal membrane. Then within the cell, you synthesize thyroglobin, right? Then thyroglobin and iodides, they are thrown through the apical membrane or luminal membrane to the colloidal area, right? Luminal area, where with some synthetic important steps are done, and then T3 T4 as a part of thyroglobin is stored within the colloid. And when we need T3 T4 in the blood, of course, if we need T3 T4 in the blood or for the peripheral tissue, we have to take from the colloid to back to the blood. If we have to take it from the colloid back to the blood, then it has to be taken, yeah, to the cells back by the endocytic process. From here, we will make the T3 T4 pull out of the thyroglobin structure, and eventually T3 T4 should come into circulation. So it means how many steps are there? There's 7 steps in synthesis, storage, and release of T3 T4. Let's remove one cell and go into detail of the function of this one particular cell. I'm going to remove this cell and put it over there and go into detail of its function. Let's suppose this is the basal membrane, base of the cell, right? This is its which part? Very good, lateral. And what is it here? Apical part or luminal part. Is that right? Now, I've removed this cell over there and we'll see how it is functioning. And of course, as good student, you won't forget that there are other cells also on the side of it, right? For example, I can make other cells just to show you, so that now yeah, now these are follicular cells, and these follicular cells have central area in which colloid has to be collected. And here is your circulation. Here is your circulation in thyroid gland. Now we'll concentrate on the function of this particular cell. For synthetic purposes, it needs raw material. One of the most important raw material which is needed by the cell is iodides. You know, iodides are coming from GIT. Iodides are normally present in our diet. From there they appear into our circulation. Iodides appear into circulation, and of course from our circulation they spread all over the body and into interstitial area. Remember one thing, all the iodides which are daily absorbed from the GIT, about 20% is usually taken up by the thyroid gland or thyroid follicular cells. 80% of these iodides are lost into urine. Again, let me repeat it. The iodides is normally present abundantly in our diet, but in some areas it may not be present in sufficient amount. That is why in many parts of the world table salt is iodized, in which one part of iodine is present in 100,000 part of the salt, right? So that there should not be deficient supply of iodide to the population, right? Let's suppose if you are taking enough amount of iodides in your food, iodide will be absorbed from GIT in the same way as chlorides are absorbed, and eventually iodides will appear in your circulatory system. Out of circulatory system, about 20% of the whatever iodides are present in circulation daily, whatever iodides are coming from GIT to circulatory system daily, about 20% is taken up normally by the thyroid tissue. Which part of the thyroid tissue? Follicular cells, right? And 80%, you can say 20% is going to the thyroid tissue, and about 80% will eventually go to the renal system, and from there it will get lost into urine. Is that right? Now, this iodide, how it will come into cell? You know, iodide is basically charged molecule, and charged molecule or polar compound cannot dissolve into lipid, so they cannot enter directly into cell. So iodide has to be taken by a special mechanism from the interstitial area into the cell. For that purpose, there is a very special pump over here, the very special pump over here, and this pump, right, this is active transport mechanism. We also call it iodide trapper or iodide pump. These iodide pumps are specially present in basal membrane. Iodide pumps are not present on the lateral membranes. Iodide pumps are not present on the apical membrane facing the lumen. Iodide pumps are present where? Again, will you help me to remind it? Yes? Yeah, iodide pumps are present on the basal membrane, right? Because they have to take iodides from the circulation or interstitial area into the cell. As I already told you, that iodides are basically charged molecule. They cannot dissolve. So they have to be taken into the cell, right? And when they enter into the cell, they are taken by active transport mechanism. They are taken up by, yes, active transport mechanism, and iodides are concentrated into the follicular cells against the concentration gradient and even against the electrical gradient. As you know, these cells do have resting membrane potential. These cells are also electrically negative inside, right? So they, if negative ions have to be pushed into cell, they are moving against the electrical gradient. In the same way, iodide is less concentration in the blood and higher concentration into these cells. So it means they are concentrated. Yeah, they are concentrated into the cell against the electrical and chemical concentration gradient. Once the iodides are present within the cell, right, they will diffuse down through the cell towards the luminal side. Iodides will move towards the luminal side. When iodides are moving towards the luminal side, let's suppose these are iodides, they are moving towards the luminal side, right? There is something extremely important, that there is an enzyme present over here, and this enzyme, yeah, what is the name of this enzyme? Yes, what is the name of this enzyme? Okay, let me tell you the duty of this enzyme first. The duty of this enzyme is that as iodides are passing through this membrane, when they appear here, they are converted into iodine. They are converted into iodine. Is that right? And in this process, they use hydrogen peroxide. It means this enzyme is leading to oxidation of iodides. Iodides are oxidized and converted into iodine. So what is the name of this enzyme? This enzyme is called peroxidases. The name of this enzyme is called peroxidases. Okay, let me tell you what are the different functions of peroxidases. Again, peroxidases are special type of enzymes, Right. So we were talking about a very important enzyme, that is thyroid peroxidase. Now, thyroid peroxidase has many functions. One function of thyroid peroxidase was that it was converting, yes, it was converting iodides and oxidizing them into iodine. It was converting them and oxidizing them into iodine. Is that right? This was one function of this enzyme. Now, this enzyme is basically present and attached with the apical membrane, right? And because it is attached with the apical membrane, right, it has very unique way of functioning. This enzyme has a very unique way of functioning. Number one, yes, number one, it is leading to this function number one. What is the function number one? It is leading to oxidation of iodides into iodine. This is one function, but it has some other functions also. For to discuss those functions, we have to come back to the cell, right? Cell is also involved into one more function, that is the formation of unique type of protein called thyroglobulin. Again, how thyroglobulin is formed? It's too easy to understand that here is the nucleus, and in the nucleus, you know, there are genes, right? In the thyroid cells, a special type of gene is activated, and when this gene is activated, this produces a messenger RNA, and this messenger RNA, when it is translated, of course, through the ribosomes, again, there is a unique type of gene present over here, and when this unique type of gene expresses itself, it makes a unique type of messenger RNA. And this messenger RNA, when it is translated, it makes a unique type of protein. And this protein, which is synthesized by this messenger RNA, with the guidance of this messenger RNA, this protein is called thyroglobulin. What is the name of this protein? Thyroglobulin. Like other proteins, it must be a chain of amino acids, right? There are many types of amino acids which are incorporated into this, but the point is, the many amino acids, but one very important amino acid is tyrosine, right? Then there may be another tyrosine. Then there are some other amino acids, and again there is a tyrosine. Of course, this tyrosine is like a tire. Tire means it is like a ring of carbon. Then again there may be tyrosine residue. So what we really see? What we really see that these follicular cells are activating such genes which make such type of messenger RNA that when these messenger RNAs are translated, they eventually end up in formation of a protein, and this protein is called, yes, thyroglobulin. Of course, this protein, once it is synthesized, right, once this thyroglobulin protein is synthesized, one thyroglobulin protein has many tyrosine residues. It has many tyrosine residues, right? This protein will be pumped in endoplasmic reticulum, right? And once this protein is going into endoplasmic reticulum, right, when this protein will enter into endoplasmic reticulum, it will get there glycosylated, so it will convert into glycoprotein. From there, from the endoplasmic reticulum, this will be packed into small vesicles, and through those vesicles it will be released towards the, moving towards the, yes, apical side. On the way, it has to be finally packaged through which operation? What is the structure? Golgi apparatus. Again, let me tell you, step number one here, gene is activated. Step number two, messenger RNA is synthesized. Step number three, messenger RNA translated into a protein. This protein is a very unique protein because in the amino acid sequence, a lot of tyrosine is incorporated into this structure. For example, this is a tyrosine, there may be a tyrosine, there may be a tyrosine, there may be a tyrosine. So we can say this protein is rich in tyrosine. This tyrosine rich protein is taken up by endoplasmic reticulum. There it is further modified and glycosylated. Then eventually it appears into what? Golgi apparatus. There it is finally packed, and eventually it is ready to be secreted from apical face of the cell, right? So we can say that through the apical face of the cell, right, protein which is packed into this particular vesicle, right, this vesicle will fuse with the apical membrane, and eventually this protein will be released in where? Into luminal area. Now let's suppose I show this protein here. This is that protein, right? As you know, it has many amino acid, different type of amino acids, right? The point which I want to highlight, that there may be many types of amino acids which are present over there, but the most important amino acids which are relevant with us right now are your, which one? T3, so tyrosines, which are important for you, tyrosines. Is that right? Now, this amino acid is different, this amino acid is different, this is tyrosine, this is tyrosine, again this is tyrosine, here it is another tyrosine, some other amino acids, again you end up with tyrosine and another tyrosine. So what we really see in this whole protein structure, which is called thyroglobulin, many tyrosine amino acids are incorporated, many tyrosine structures are incorporated. Now, as you know, that one function of, what was this enzyme? What was the name of this enzyme? Please tell me. Peroxidase. Which peroxidase? I'm not very good at drawing. Please, no one should mind it. Right? And don't think it has anything to do with anyone very near and dear to you. This is tyrosine peroxidase. So one function is, it is converting what? Iodides into iodine. Other function which is there is that once, what is the structure? Thyroglobulin. Second function of this protein is, okay, I think it's not good. Let's make it as plain as possible, right? Some people may feel uncomfortable with this type of situation. Now, second function of this enzyme is that it will fuse this iodine with the tyrosine residues. It will fuse these iodine with tyrosine residues. Now, due to the second function, what really happens, that we say that due to the second function, the iodine, which is inorganic compound, when it becomes associated with tyrosine, we say iodine is organified. Iodine is organified. So, iodine may bind here, or at this tyrosine, iodine is binding at 2 points. Now listen, when iodine is binding at one point in the tyrosine, we call it monoiodotyrosine. What is this function called? Monoiodotyrosine. So monoiodotyrosine. On this tyrosine, the next tyrosine, we have added how many iodides? 2. So because 2 iodines are there, we call it diiodotyrosine. It means one another function of this particular a very important function of this particular enzyme was not only to convert iodides into iodine, but help the iodine to get organified by fusing with the tyrosine. So this is its second function. Is that right? That it has led to the, what was this function? Organification. And this was its first function. What was it? Yeah. Now we have seen two functions of this enzyme. One function is iodides into iodine. This is peroxidase function. Is that right? This enzyme is thyroid peroxidase. Second function was, it was incorporating iodine with tyrosine, and when it was incorporating iodine with tyrosine, it was leading to organification of iodine. Second function. And when it will repeat the same function with multiple thyroids present over there, multiple tyrosines present over there, right? Eventually what will happen, that multiple mono and diiodotyrosines are formed. For example, here is again maybe one iodine, then another iodine. Here is iodine. It means they have double iodinated. If this tyrosine is double iodinated, this is diiodotyrosine. This tyrosine residue is also double iodinated. It is also diiodotyrosine. Is that right? Here, let's suppose again there is only single iodination, and here there is double iodination. And again we'll repeat it. Here it is double iodination. Here is also double iodination. So it means that this enzyme has led to oxidation of iodine, iodides into iodine. It has also led to organification of iodine, and it has one more very important function. You know what is that? It helps these two adjacent iodinated tyrosine to fuse with each other. Right? How? This is another function. Very important function over here. What is the other function of this? Other function of this enzyme is it helps these two adjacent to fuse with, yes, to fuse with each other. And if they fuse with each other, this, it means they are making a couple. So this is called coupling reaction. It means monoiodotyrosine, and here it is diiodotyrosine, they are coupled with each other. And when they are coupled with each other, it means that within the structure of thyroglobulin, first two iodine residues were iodinated, and after they were loaded with iodine, they are fused with each other, right? And if they are fused with each other, what really happens? Monoiodotyrosine fused with diiodotyrosine. When monoiodotyrosine, right, fuse with the diiodotyrosine, then both of them together make triiodotyrosine. Triiodotyrosine. Again, let's focus on it and concentrate this concept. What was going on at this point? That here it was, what was it? It was tyrosine residue. This was also a tyrosine residue. Is that right? And this first tyrosine residue, it was having one iodine, and second tyrosine residue, it was having how many iodine? Two iodines. If first was called monoiodotyrosine, second was called diiodotyrosine, right? And if they are fused together, right, then they are called triiodotyrosine. Similar reaction may occur in some other parts of thyroglobulin also. Let's suppose focus on this point. Let's suppose focus on that point. Let me explain it with more clarity. Let's suppose there is another tyrosine residue here, and there is another tyrosine residue here, right? And let's suppose in this case this is double iodination, this is also double iodination. So naturally this residue should be called diiodotyrosine. This should also be called diiodotyrosine. Is that right? Now the point which we have to understand, that if this magical enzyme also lead to coupling of this, right, they are really very happy. You know why? They are going to, yes, couple. Do they look to you as a male or females? Yes, what do you think? They are both males or females? No, please let me know. Is it males or females? Let's focus on that and see. Yeah, you want the male and a female, but they look like each other. So there are two females or two males. Okay, let's come like this. You're laughing. You don't want to reveal your unconscious to me. Okay, fine. So what I was talking about, actually, they are not male or female. They are just tyrosine loaded with iodines, right? Now it has diiodotyrosine. This is also diiodotyrosine. Again, this enzyme come into action. And if this enzyme is coming into action, right, and if it lead to coupling of that, right? If it leads to, what is this? Coupling of that. And if both of them are coupled, now diiodotyrosine fuse with another diiodotyrosine. Now this unit should be called tetraidotyrosine. Now you see the beauty of understanding that this was one single thyroglobulin molecule, and within the thyroglobulin molecule there were too many tyrosine residues, and these tyrosine residues were getting iodinated, and once they iodinated, initially they lead to the formation of monoiodotyrosine and diiodotyrosine. Within this whole structure, there are many monoiodotyrosine and there are many diiodotyrosine. Then what happens? This enzyme will lead to coupling reactions. Is that right? When this enzyme will start coupling reactions and stitching together, of course biochemically, adjacent iodinated tyrosine residues. If monoiodotyrosine fuse with diiodotyrosine, you end up with formation of T3. And if diiodotyrosine is fused with nearby, coupled with nearby diiodotyrosine, you end up with a structure, combined structure called T4, thyroxine. It means T3 and T4 are formed within the structure of thyroglobulin. Now look, we come over here. There are some more amino acids here, and again what is this? Tyrosine and another tyrosine. These are two tyrosine. If I say it is only iodinated at one point and other one is iodinated at 2 points, then what is this? Yes, monoiodotyrosine, and this is diiodotyrosine. Is it right? Now let's suppose this enzyme ignore this unit and this mono and di are not coupled. Mono and di are not coupled. It's quite possible in your life you come up with many people who are never coupled. At least they claim that they have never coupled. Right? Now we can come to again some more thyroglobulin unit. Again, this is, what is this? This is again tyrosine residues, and let's suppose here is iodine, double iodination here, and here is also iodine. Now what is it? Diiodotyrosine and again diiodotyrosine, and if thyroid peroxidase couples them together, right, what will happen here? Formation of T4 thyroxine. It is T1 and this is T2, theoretically speaking. It means this is one peptide chain in which tyrosine residues were first attached with iodine, and there was formation of monoiodotyrosine and diiodotyrosine. Then the many points they were coupled with each other, and due to coupling reaction again by the same enzyme, what was the result? Yes, T3 formation, T4 formation. So we can say T3 formation and T4 formation is within the molecule of thyroglobulin. Is that right? So we can show this molecule in a more simplified way like this. We can show it like this, and we can say here, look at special points, it is T3, here it is, yes, what is it? T4, here it is T1, here it is T2. Of course T1 should be MIT and T2 should be called DIT, right? And there's so many such units here. So what is this? This is one thyroglobulin, this is another thyroglobulin, this is another thyroglobulin. So there is a lot of thyroglobulin present over here, and all this thyroglobulin is having with as a part of it T3, T4, T1, T2, T3, T4, T1, T3, T4, 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 T1, T2, T3, monoiodotyrosine T1, diiodotyrosine T2, T4, thyroxine, triiodotyrosine T2, T1. So it means that all these cells which are present on the side, they are secreting thyroglobulin into lumen. They are secreting thyroglobulin into lumen, and iodine is binding with the tyrosine residues, and then coupling reaction is occurring, and T3, T4 are being formed and synthesized as a part of thyroglobulin molecules. And when many thyroglobulin molecules are together present into the follicle, all this proteinaceous, glycoproteinaceous material which is heavily iodinated. This material is called colloid. Very good. This material is called colloid, right? So what is colloid? Colloid is glycoproteinaceous material present in the lumen of a follicle, and this material is secreted by the follicular cells through their apical membrane. And within the colloid, it is having a lot of thyroglobulin, and these thyroglobulins are having residues of monoiodotyrosine, diiodotyrosine, or triiodotyrosine, or tetraiodotyrosine to thyroxine. Is that right? Any question up to here? No. Again, let me repeat it. This enzyme, very important for us. This enzyme, how many functions it was doing? Yeah. Number one, function number one was yes, oxidation of iodides. Function number two, what was it? Organification of iodine. Function number three, yes please, coupling reaction. So now you know the three functions of this enzyme. One, two of both arm are busy, one leg is busy, and one leg is resting. And again, what is the name of this enzyme? Yes, thyroid. What is the name of this enzyme? Thyroid peroxidase. Peroxidase. The name of this enzyme is thyroid peroxidase. So it is extremely important enzyme doing three important functions. What are those functions? It leads to oxidation of iodine, it leads to organification of iodine, attachment of iodine with tyrosine, it leads to coupling reaction. Any question here? No. Now let's continue. Now we know that T3, T1, T2, T3, T4, MIT or DIT or T3 or T4, they are part of what? T3, T4, they are part of T3, T4, MIT and DIT. These are part of thyroglobulin in the colloid. I want to tell you something very important. If there is good supply of iodides, we'll make more T4. It's very understandable. If you have abundant supply of iodides, then it means you will do double iodinations. It means you will make more diiodotyrosines and less monoiodotyrosine. It means you will synthesize more T4. Is that right? But if there is deficiency of, listen carefully, if there is a deficiency of iodides in your blood and less iodides are coming here, then what happens? That monoiodotyrosines are made more, diiodotyrosines are made less. So net result is T3 is made more. Normally in thyroglobulin, attention please, look here. Normally what really happens that about 93% of the T3 T4 which is released, 90%, 90 to 93% is T4 and only 7 to 10% is normally T3. It means T4 is released more and T3 is released less. But again I would like to repeat that if iodine is abundantly present, right, synthesis of T3 is very less. But if iodide supply is limited, then synthesis of T4 slightly reduces and synthesis of T3 slightly increases. Right? Now, let's suppose next point is that the cell has taken raw material from the blood. As we know, the cell has taken iodide from the blood. This cell has taken tyrosine from the blood. It has taken many other amino acids. Iodides are converted into iodine there, and many amino acids are eventually incorporated into thyroglobulin, including very special amino acid, your friend tyrosine, right? Then we synthesize T3, T4 as a part of thyroglobulin and dump into colloid. Is that right? But the question is that when your body needs T3, T4, we have to get it back into blood. Now, now listen carefully. Here is the point where thyroglobulin is present and T3, T4 is incorporated within thyroglobulin, but actually you want T3, T4 in the blood. Now, how from the lumen it can come to the basal side? Answer is very funny, that when the cell want to synthesize and store the T3 T4, reactions are going into this direction. And when the cell want to release the T3 T4, reaction has to move from the lumen side to basal side. What really happens? Actually, when the cell intend to, right? What the cell is going to do? Okay, I think this is too much exaggeration. Okay, let me make a smaller look. What the cell will do? The cell will make pseudopods. The cell will make pseudopods. And these pseudopods will move like this, and they will engulf a chunk of colloid. They will take up a chunk of colloid. And when this chunk of colloid will become part of it, right, then they will move, invaginate inward and close it. What is the result? The result is that from the lumen, a piece of, what is this? Colloid, of course, thyroglobulin, right? It has been under pinocytic process. And through the pinocytic process, it has taken a chunk of colloid back into cell. Now this is intracellular, right? This is pinocytic vesicle. In pinocytic vesicle, what it is having? It has monoiodotyrosine. It is having, yes, diiodotyrosine. It is having T3 and it is also having T4 and many other amino acids as well, right? Let's keep on focusing on this, that this was a pinocytic vesicle and in which there is a small piece of colloid. Basically, this is what? This is thyroglobulin, and in this thyroglobulin there are monoiodotyrosine, diiodotyrosine, triiodotyrosine, tetraiodotyrosine, and some other amino acids as well. Some other amino acids are also present. Is that right? Now, what we do once we have engulfed it, we have to digest it. For digestion purpose, there should be some lysosomal enzymes present in lysosomes, and these lysosomes will be fused with it. And when these lysosomes are fused with it, lot of lysosomal enzymes are pushed into this pinocytic vesicle. And now this pinocytic vesicle will convert into digestive vesicle. Now in this digestive vesicle, what really happens? Right, we have to see this lysosomes are fused there, and in this digestive vesicle, what really happens? Already you know there are many amino acids, including the tyrosines and T3 T4. Let's suppose here it is monoiodotyrosine, here it is diiodotyrosine, here it is T3, again there is T4, and then again there is T4. Now look, this was T4, this was T4, here it is T3, this is diiodotyrosine, here it is monoiodotyrosine. And what is this funny enzyme? It has a cutting point. What is the name of this enzyme? It is proteolytic enzyme. What is the name of this enzyme? Yes, please. Proteolytic enzyme. It came from where? From lysosomal system. This proteolytic enzyme will start cutting down the peptide chain. When this will start cutting down the peptide chain, there will be release of, yes, release of monoiodotyrosine, diiodotyrosine into cytosol, T3, T4, again more T4. And of course, don't forget many other amino acids. Now out of this, T3 will slip into circulation, right? And of course, T4 will also slip into circulation. Actually, normally, T3 is released in circulation in smaller amounts and T4 is released in circulation in larger amounts, right? So it means that normally when T3 and T4, which are combined called thyroid hormones, T3 is released in smaller amounts and T4 is released in larger amount. Of course, we will know T3 is more active form and more rapidly active form, and T4 is slowly acting form, right? We'll talk this thing later. But one thing which I want to highlight, this is what? Diiodotyrosine and monoiodotyrosine. They will not go out of the cell. They will stay within the cell. And what really happens? Suppose this was your tyrosine residue. This was diiodotyrosine. Right? Monoiodotyrosine. Okay. Monoiodotyrosine, there was single iodination, and diiodotyrosine, there was double iodination. Now what really happens? This MIT and DIT, they are not allowed to go into blood. They are not allowed, right? They are not allowed. They will turn back. But T3 and T4, of course, you now know the structure of T3. Let me draw here for you the structure of T3. Let's focus on that. This is the structure of, yes please, T3. This is tyrosine residue. This is also tyrosine residue, right? And the one iodine here at position number 3 and 5, iodine here, and there should be, okay, if it is T3, there's one iodine here and there are two iodines here. This is T3. But when we compare the structure of T3 with T4, this is structure of T4. You see it has how many iodination? At both tyrosine, double 3 and 5 iodination. Now you see. In T1 it has only, or you can say monoiodotyrosine has only one point where it is having iodine. Diiodotyrosine, it has 2 points where it has iodine. At 3 there are two tyrosines which are coupled, and one is having one iodine, another is having two. And when we talk about T4, basically two tyrosine residues are coupled and both are having double iodination. These double, which are making T3, they are allowed to go into circulation. This doublet, which is making T4, is also allowed to move into circulation. But this singlet and this singlet, they are not allowed to move in circulation. Rather, what happened? A very special enzyme is waiting for them. There is a very special enzyme which is waiting over there, and this enzyme, what this enzyme is going to do? It will pluck out their iodines. It will take up their iodine and pull that away. So what it is going to do? It is going to remove the iodine from there. It means it is doing deiodination. What is the function of this enzyme? Deiodination. So this enzyme is called, it is a strange type of cap. It is written here, thyroid deiodination enzyme. What is the name of this enzyme? Thyroid deiodination enzyme. This enzyme is very important because congenitally in some people this may be deficient. And if this is deficient, then iodine cannot be removed from monoiodotyrosine and diiodotyrosine. And if cannot be removed, then it cannot be recycled, because this iodine, once it is removed, okay, let me change the color of this enzyme so that you can appreciate it better. The function of this enzyme is what? To pull this iodine away, right? To pull this iodine away. And once it removes this iodine, what really happens to this iodine? This iodine is going to the back to the pool to be reused. Did you get it? Now, if someone has deficiency of this enzyme in his family, inherited defect, the genes which are concerned with synthesis of this enzyme, if those genes are not working well, an enzyme is not present, or if it is present and not functional, then from monoiodotyrosine or diiodotyrosine, we cannot remove the iodine. If we cannot remove the iodine, we cannot recycle it. If we cannot recycle it, there will be deficiency of iodides and iodine, and eventually result will be poor formation of T3 T4. 4. Is that clear? Again, let me tell you something. Right? Let's recap all of it. Let's recap very simply. We said that we are going to talk about synthesis of thyroid hormone. When we say that we are talking about the synthesis of thyroid hormones, it means we are talking about the synthesis of T3 and T4. Is that right? Now, where T3 and T4 are synthesized, everyone knows. They are synthesized in thyroid gland. In which part of thyroid gland? In the follicular cells and the lumen of them. Within the follicular cell and into lumen of them, right? Remember, T3 T4 is not synthesized by parafollicular cells because parafollicular cells in the thyroid gland are concerned with the synthesis of calcitonin. Please don't confuse that. So what are follicles? Follicles are special balls of functional unit, spherical functional unit made of cuboidal cells within thyroid tissue. Is that right? Now, we have removed one cell and we are looking at how it functions, right? How it makes T3T4. Actually, we have to see how it makes T3T4, how it stores T3T4, and how it releases T3T4. Is that right? And the first step, first of all, we should remember that this cell has a basal side which is towards the capillaries and interstitial area, and this cell has one luminal side which is facing to the lumen of the follicle where lot of colloid is present, glycoproteinaceous material. Clear? Now, first step should be that you should take up the raw material like iodides and tyrosines. Number 2, within the cell it should synthesize thyroglobulin. Is that right? Number 3, iodide should be thrown into lumen. While they are passing through that, this enzyme will attack. Peroxidase will attack and convert the iodides into iodine. Meanwhile, when thyroglobulin is synthesized during the translational process, lot of tyrosine residues are incorporated within the thyroglobulin. Thyroglobulin molecule is glycosylated within endoplasmic reticulum, stored into vesicles, packed into Golgi apparatus. Again, through the apical vesicles, it is released into what? Lumen. In the lumen, here we have shown a typical thyroglobulin. This typical thyroglobulin has many amino acids, and out of them, some of them are tyrosine residues. As soon as thyroglobulin is passing into lumen, right, iodine, which is already there, right, with the help of the action of thyroid peroxidase, this iodine will start attaching with the tyrosine residues. If at one point if it attaches, it is monoiodotyrosine. At 2 points it attaches, it is diiodotyrosine. Right? So many monoiodotyrosine and diiodotyrosines are found within the thyroglobulin. Then this enzyme has another action. You know what is that action? That this enzyme, which has already done the oxidation of iodides and converted them into iodine, and then second step this has done, it has led to the organification of iodine. Third step, which is it is going to do a very pleasant job, something like a good pastor. It will do help them into coupling. Monoiodotyrosine will fuse with diiodotyrosine, or of course you can say diiodotyrosine may fuse with monoiodotyrosine, and any one of these reaction will end up with the formation of T3, triiodotyrosine. The same way, if diiodotyrosine fuse with another adjacent diiodotyrosine, there will be formation of T4. If T3 and T4, right, they are formed as a part of thyroglobulin molecules. They are formed as a part of thyroglobulin molecules. Now, within the thyroglobulin molecule, many T3, T4 are there, but many monoiodotyrosine and diiodotyrosine are still there, which are uncoupled. Which are never coupled. Right? Now, whenever we need to, you can say secrete T3, T4, these cells from the luminal side will start pinocytosis, and this apical surface of the cell will start pinocytic pseudopods, and they will take up the chunks of the colloid, and colloid will come into this pinocytic vesicle, and of course colloid is having a lot of thyroglobulin, and this thyroglobulin is having T1, T2, T3, T4. Lysosomal enzymes are added to that, and proteolysis start. Another enzyme here is thyroid proteolytic enzymes, and they will break down the thyroglobulin into amino acids, plus there will be release of T3 T4, which will be released into circulation. T3 is released in smaller amount and usually T4 is released in larger amount. At the same time, that monoiodotyrosine and diiodotyrosine, which was not coupled, right, that is also released into cytosol but not into circulation, right, and their iodine has to be recycled, so iodine need to be pulled out of these molecules. The function is done by this enzyme thyroid deiodinase, and it will remove these iodines and let them recycle and to be reused. Is that clear? Let's have a break. Now we are going to talk about antithyroid substances very briefly again, right? In detail, we'll talk about these substances in pharmacology. First of all, some of the antithyroid drugs or substances work on the iodide pump, right? For example, there are substances called thiocyanate, or there are perchlorates, right? Thiocyanates and perchlorates, these are anions. They are also anions like iodides. Basically, these thiocyanate and perchlorates, they competitively inhibit the uptake of iodides from the iodide transporting pump by using this pump for themselves. For example, if I say that here is thiocyanate, now what is the function of thiocyanate? What this thiocyanate is doing? Number one, it is trying to go up, right? Itself, it is trying to go up into and using this pump for its own purpose. Secondly, a very important thing, it blocks the entry of iodide to the, and iodide is repelled back. So what really happens? This is how thiocyanate and perchlorates work. The thiocyanate or perchlorate utilize these iodide transporters on the basal membrane for their own purpose, and they antagonize the entry of iodide through these pumps into thyrocytes or into thyroid cells, right, or follicular cells. It means eventually what is the result? The result is that these cells become iodide deficient. These cells become iodide deficient, and when these cells become iodide deficient, naturally iodides are less, and what will be the result? That eventually iodine formation will be less. It means T3 and T4 formation will be less, and if T3 T4 formation is less, all the results, storage is less, its pinocytosis is less, its proteolysis is less, and T3 T4 release in circulation is less. So it means the result of using thiocyanates or perchlorates is that follicular cell cannot take up the iodides. When they cannot take up the iodide, they cannot synthesize properly T3 and T4. So there's deficiency of T3 T4. As you know that if there's deficiency of T3 T4, then this cannot inhibit the production of TSH. So as T3 T4 levels are going down, TSH production by the interior, what is this? Interior pituitary, the production of TSH will increase. And when production of TSH will increase, as we will study shortly, that TSH level will go high and they will overstimulate TSH receptors. When TSH receptors will be overstimulated, they will give signal. Number one, to increase synthesis of T3 T4. Number two, they will give signal for hypertrophy or enlargement of the follicular cells. And number three, they will give signal for proliferation of follicular cells. Now listen, what will be the net result? Again, if we have a patient here, what is this drug? Thiocyanate or perchlorate. It does not allow iodide to enter into these follicular cells. When iodides cannot enter into these follicular cell, iodide deficiency eventually result in iodine deficiency, eventually result in reduced formation of T3 T4, and eventually reduced production of T3 T4. When T3 T4 is produced less, what is the result? That TSH cannot be kept suppressed as T3 T4 levels are going down, especially when T4 levels are going down, level of TSH in the blood increases, right? When level of TSH in the blood increases, TSH receptors on these follicular cells are stimulated, right? They try to increase they give stimulation to increase the synthesis of T3 T4, plus they give signal for increasing the size of follicular cells. Number 3, they give signal for proliferation of follicular cells. Now, as iodide deficiency is there, the synthesis of T3 T4 cannot be increased. But increasing TSH eventually lead to increased size of follicular cells and increased proliferation of follicular cells, which eventually result in enlargement of thyroid gland. And when thyroid gland is enlarged, it means there is formation of goiter. What is this? There is formation of goiter. So it means that when you are using thiocyanate and perchlorate for longer time, you are achieving two results. One result is reduced formation of T3 T4. Is that right? It means they are anti-thyroid drugs, right? Function of the thyroid tissue is reduced. But inadvertently, when T3 T4 level is less, unfortunately TSH level goes up. TSH start overstimulating the thyroid follicular cells. Even though due to deficiency of iodides, synthesis of T3 T4 cannot be increased, but size of follicular cells and proliferation of follicular cell is increased, that result in enlargement of thyroid gland, and we say that there is formation of goiter. Is that right? Now, after this, there is another group of drug rather, which can inhibit the function of thyroid peroxidase. Now, what are these drugs? Do you know any drug like that? Propyl thiouracil. Propyl thiouracil. What this drug is doing? Propyl thiouracil, right? That is basically inhibiting this thyroid peroxidase, right? Let's suppose I make here propyl thiouracil, right? This is propyl thiouracil, right? What it is doing? Look at it, how it is working, propyl thiouracil, right? Now, what it is doing? It's a very unfortunate activity. It is eating up the function of, yes, it is disturbing the function of thyroid peroxidase. This is disturbing the function of thyroid peroxidase. Is that right? Because it disturbs the function of thyroid peroxidase, so naturally thyroid peroxidase will be working less, right? What is the name of this? Propyl thiouracil, and it has its friends also. The friends of this are carbimazole and, yes, methimazole, especially carbimazole or propyl thiouracil. They have similar action, that these drugs basically inhibit the action of thyroid peroxidase. I think better way to draw these drugs is that even they love to, yes. This is again which drug? This group is the same group, propyl thiouracil, propyl thiouracil or carbimazole, right? So what are the function of propyl thiouracil or carbimazole? They are basically inhibiting the function of thyroid peroxidase. When they inhibit the function of thyroid peroxidase, what is the result, right? The result of that activity is that, yes, the result of this activity is that you cannot convert iodide into iodine, and you cannot, if there's any iodine present, you cannot utilize to convert tyrosine residues into monoiodotyrosine or diiodotyrosine, and whatever monoiodotyrosine and diiodotyrosines are there, you cannot do the coupling reaction to lead to the T3 T4 formation. Eventually, by using such group of drugs, you also lead to reduced formation and release of T3 T4. And again, what will happen? That there will be excessive release of TSH by anterior pituitary, and when there is excessive release of TSH by the anterior pituitary, that will lead to overstimulation of these follicular cells for the increased synthesis of TCT4, but increased synthesis of TCT4 will fail due to deficient function of thyroid peroxidase, but there will be increased size, increased proliferation of these cells, and eventually again formation of a goiter. So what did we learn? That if you use thiocyanate or perchlorate, by reducing the uptake of iodide, you reduce the T3T4 formation, but TSH will be increased, goiter may form. Second example we have taken, if we use thiopropyl uracil or carbimazole type drugs, they inhibit the function of thyroid peroxidase, and if thyroid peroxidase is not working, you know that iodides cannot be oxidized, then iodine cannot be organified, and then coupling reaction cannot be done, and result is that again TCT4 is less and TSH is more, and again to increase TSH will overstimulate these cells to produce more T3T4, but increased production will fail, right? They will also give stimulation for increased size of the cells and proliferation of the cell, and that may occur, that may lead to the formation of goiter, right? So it means these anti-thyroid drugs which we have discussed up to now, with prolonged use, they may lead to formation of goiter. Goiter means enlarged thyroid gland. Remember one thing, goiter does not tell about if I say your patient has goiter, it does not tell what is his functional status. Goiter just means that size of the thyroid gland is more, right? But it does not tell that is this patient hyperthyroid or euthyroid or hypothyroid. But if you are using these drugs, of course patient will eventually become, if it was a hyperthyroid patient, by use of these drugs patient may become euthyroid or even hypothyroid. Is that clear? Now we are going to talk about a very special type of drug. Sometimes just before surgery, few weeks before surgery of for the thyroid gland, we give the patients potassium iodide, excessive amount of iodides. Now we must know the relationship of this pump with the iodides. Okay, let me show it here, right? That this is another iodide transporter, right? It is another iodide transporter, and what is the relationship of function of this transporter, iodide transporter, with the iodide concentration? Remember one thing, if in your body iodide is less, its function will become more, so that for reduced amount of iodides, it can compensate by increasing the function, so that out of less iodide it can concentrate enough amount. But if you keep on increasing iodide concentration in your body or in your blood, it will become gradually slow. So again, this pump is really very intelligent. Its main purpose is to keep a good, you can say, concentration of iodides and good supply of iodide to the follicular cells. If in the body iodides within physiological limits, if iodides become less, this will become more active. So out of less concentration of iodide in the interstitial area, it can still take enough amount of iodide in the follicular cell. And if there is excessive amount of iodides, this pump will become slow, so that it will still, out of too much iodide, it will take only that amount of iodides in the cell which is just enough. But if you increase the iodide concentration too much, if you increase the iodide concentration too much, for example, you feed your patient on very high concentration of iodides, right? Let's suppose here is your patient, and this patient has, you have provided him with lot of iodides. What will happen when there's too much iodide in your body, right? What this iodide will do, all the iodides will lead into, yes, excessive amount of iodide will eventually lead to stimulation of this pump or inhibition of this pump. Yes, it will lead to inhibition of this pump. So eventually this pump will fail. This pump will fail. So it means one way to inhibit your iodide trappers is give so massive amount of iodides that this pump is overwhelmed, right, and it is inhibited. It looks really very paradoxical, but it is true that if in your body, first listen the basic concept, if iodides are going down, pump is becoming more effective. If iodides are becoming more, pump is becoming less effective. If iodides become very high, pump become ineffective. So it means one way to reduce iodide uptake is provide lot of potassium iodide orally, and it should go to absorb and go to your circulation, and massive amount of iodide will inhibit this pump, right? And when this pump is inhibited by massive amount of iodides, right, what will happen? Again, T3 T4 formation is reduced. What happens? T3 T4 formation is reduced, and you can say it acts as anti-thyroid mechanism. But there is one different thing. Previously we discussed, when you use thiocyanate or perchlorate, or you use propyl, thiouracil or carbamazepine, right, all of them lead to increased TSH level and eventually may lead to goiter formation. But the good news about this type of treatment is that it does not lead to goiter formation. Rather, it's a very important point. Please remember that this leads to reduced size of thyroid gland. Reduced thyroid gland, yes, function and size both. Function and size and vascularity. It means few weeks before surgery, if you give the patient potassium iodide, right, patient's gland will shrink and its vascularity will become less, and it will become less hyperfunction, or it may become hypofunction. Especially it will inhibit immediately proteolysis, pinocytosis and proteolysis, and further release of T3 T4. That is why frequently potassium iodide is used before surgery, so that gland should become smaller in size, it should become less vascular, and it become less functional, so that surgery become easier. Is that okay? Any question up to this? Right? This effect, that excessive iodide inhibit the iodide transporters, right? This effect is called Wolff-Chaikof effect. What is this effect? Wolff-Chaikof effect. It means when iodide become excessive, they behave like a wolf and bite it and damage it so much that they become slow into action. Is it clear? Further details of such drugs we will discuss in pharmacology. Okay, now we'll talk about that how T3 T4 are carried into circulation and how they go into peripheral tissue and where they can be further, especially T4, how it can be activated and how it can be degraded. Let's concentrate first of all on the thyroid gland. Start from here. You know, in thyroid gland there are so many follicles, and follicular cells are constantly releasing T3 and T4. T3 is released in very small amount, usually 5 to 10%. But about 90% of the thyroid hormone which is released from the follicular cell is T4, right? T4 is also called a prohormone, or because it is less active, but it can be activated further by converting into T3. Anyway, when T3 and T4 come into circulation, right, they are kept bound with special transporting proteins. These transporting proteins are constantly synthesized by hepatocytes. These are constantly being synthesized by hepatocytes, right? Hepatocytes are constantly releasing thyroid binding globulins, thyroid binding prealbumins, and thyroid binding albumins, right? But most of the T3 and T4 bind with thyroid binding globulin and little bind with prealbumin and albumin. The total amount of T3 and T4 which is present in circulation, out of that 99% is protein bound. Protein bound. It means if more than 99% is protein bound, less than 1% of the T3, T4 present in circulation is in free form. And actually this is the free form of T3 and T4 which is biologically active. Let's suppose this is thyroid binding globulin. One molecule of thyroid binding globulin has many sites where T3 and T4 can bind. Usually T4 is binding here in larger amount because T4 has stronger affinity to bind with these proteins, right? And it remains with these proteins for a longer time. T3 does bind but for a shorter time. Then what really happens? Eventually T3 and T4 are released from these proteins into circulation and they are in the form free form. Free form can diffuse free T3 and T4, they can diffuse into tissues. As you know, T3 itself is directly active. Problem is with the T4. T4 is weakly active. But T4, now here is the structure of T4. T4 can be acted upon by a special enzyme which is present in peripheral tissue. This is called 5' monoiodinase. 5' monoiodinase. The function of this is that, let's suppose this carbon number 3' and this is 5'. 5' mono deiodinase removes this iodine, right? When it removes this iodine, then T4 is converted into T3, and T3 is the form which is biologically active. Look at this T3, very happy, it is willing to work for you on the tissues. Is that right? Opposite to that, if the other enzyme work there, right, the sister enzyme is called not 5', it is simple 5 mono deiodinase. 5 mono d mono deiodinase. Now this 5 mono deiodinase, right, deiodinase. Now 5 mono deiodinase does not remove this iodine, rather this removes this particular iodine, right? And this pathway lead to this structure of T3, in which first component 3' and 5' carbons are iodinated, but 3 carbon in the other tyrosine ring remain iodinated, but 5 carbon in the other tyrosine residue is deiodinated. Now, when deiodination occurs at this point, right, this is not called true T3, rather we call it reverse T3. And reverse T3 is biologically inactive. One way to remember is, look here. In true T3, what really happens? That first tyrosine is, this is first tyrosine, it has 1 iodine, and second tyrosine has 2 iodine, 1 and 2. When we look at T4, it is first iodine has 2, first tyrosine has 2 iodinations, second iodine has 2 iodination. This is less active. In reverse form, you see it is not 1 and 2, rather first has 2 iodination and second has 1. So if you remember 2 and 1, you know 2 is coming first, 1 is coming later, it means that the pattern is reversed. In functionally active T3, 1 should be coming first and 2 should be coming later. Is that clear? So what did we learn from here? That when T3 T4 is released, Mainly it is released in the form of T4, which is pro-hormone, a storage form of the hormone. T4 bind better with the plasma proteins, especially thyroid binding globulin, lesser amount with the thyroid binding pre-albumin or thyroid binding albumin, and free form is less than 1% because more than 99% of the T3 and T4 is bound with the plasma protein. This free form keep on shifting to the tissues. T3 can be directly acting upon the cells, which I will explain shortly, but T4 itself, if it directly work on the cell, it works weakly. It has less potency. But T4 can be converted into T3 as well as into reverse T3. If there is 5' mono deiodinase, then it will convert into T3 and it will become into more active, more potent. But if it is acted upon by 5' mono deiodinase, then it converts into reverse T3, which is inactive form. Right? Now, one more important concept, as you see already, T4 is produced in larger amount and it has more affinity to bind with TBG. T3 has less affinity to bind with TBG. It means that if you give injection of T4, right, it will remain in circulation for longer time, and if you give injection of T3, it will remain in circulation for shorter time because T4 bind with protein more avidly, more tightly, so it will remain in circulation longer. T3 bind with the proteins relatively less, with less strength, so it will easily become free and go to the tissues, right? Generally, they have seen that half-life of T4 is approximately 6 days, right? And half-life of T3 is approximately 1 day. It means T3 not only I act more powerfully, it also act more rapidly. Opposite to that, T4 is released slowly by the proteins. Even though T4 is a major storage form of hormone, that is why, and it can be converted into T3, more active form. That is why it is also called pro-hormone. But because it more avidly binds with the plasma proteins, so its half-life is longer. One important concept as far as transport of these proteins is concerned is related with the function of the liver. As we know, the major transporter for T3 T4 and major binder for T3 T4 are provided by the hepatocytes. These are the proteins which are released by the liver cells. Now, in pregnancy, the synthesis of TBG is increased. TBG means thyroid binding globulin. It means in pregnancy, total T3 T4 in the blood level will also go up, right? Because in pregnant female, liver produces more TBG. If there's more TBG coming to circulation, it means there are more binding site available for T3 T4 binding. It means free T3 T4 becomes slightly less, but that slightly less T3 T4 stimulates the TSH production and maintains its free level. Right? So what we can say, in pregnancy usually total T3 T4 is more than the norm person who is not pregnant. But if we talk about the people who have problem with the synthetic function of liver, for example, the patient of cirrhosis. In patient of cirrhosis, the synthetic function of the liver has gone significantly down, and plasma protein levels are down, and TBG synthesis or thyroid binding globulin synthesis is less, then total T3 T4 level in the blood will be also less. So all those conditions which reduce the hepatic synthesis of plasma protein reduce the total level of T3 T4 in the blood, and all those conditions which increase the production of or synthesis of thyroid binding globulins and other proteins, they will lead to increased level of T3 T4 in circulation. Dear doctors, right now we are going to talk about that how the secretion of T3 and T4 is regulated through hypothalamic pituitary thyroid axis, right? So let's draw a diagram and look at it. First of all, in the central nervous system where exactly hypothalamus is present, right? And pituitary is present. Here I draw the central nervous system. Of course, this is cerebral hemisphere, this is midbrain, here it is bones, here it is medulla, cerebellum, spinal cord, right? The exact location, exact position where hypothalamus is present is this area. This is the point where hypothalamus is present, and just under it, here there's anterior pituitary and posterior pituitary, right? So this structure I'm going to isolate and draw over there, right? This is the exact position where hypothalamus is present, and just under the hypothalamus there is median eminence, and just more inferior to that there is anterior and posterior pituitary. So this structure I will draw over here. Now here is our hypothalamus. Okay, let's make it a very simple diagram. This is anterior pituitary. Here it is anterior pituitary, and here it is posterior pituitary, and there is no mammillary body over here. Now again let me repeat, this is your hypothalamus. Here is median eminence. This is an infundibulum which connect the hypothalamus and pituitary system. Here is anterior pituitary, and there it is posterior pituitary. Now how really the system works? All of you know the cells which release TSH, these cells are present in anterior pituitary. Let's suppose this is a cell, right? There are many cells in anterior pituitary which are concerned with the release of TSH. I've drawn just one cell. This cell is concerned with the release of TSH. But actually, hypothalamus releases a substance called TRH which controls the release of TSH by anterior pituitary. Now what really happens? Look here. There's a vascular connection between the hypothalamus and the anterior pituitary, and this vascular connection can collect the different hormones from the hypothalamus and median eminence and bring them into the anterior pituitary. This vascular connection between hypothalamus and anterior pituitary is called hypothalamic hypophyseal portal system. Again, let me repeat it, that there is a very special and unique type of vascular connection from the hypothalamus to the anterior pituitary, which collect a lot of substances from the hypothalamus and directly drain them into anterior pituitary, so that activity of anterior pituitary can be controlled by the hypothalamic factors, right? This vascular connection is called hypothalamic hypophyseal portal system. Hypothalamic hypophyseal portal system. Now, there are very special neurons present in paraventricular area of hypothalamus. These are very special neurons which are present in paraventricular area of hypothalamus. Now, these neurons secrete a tripeptide which is called, yes, what is the name of this tripeptide? This tripeptide is called TRH. This tripeptide is called TRH, and this TRH is coming from the hypothalamus through the hypothalamic portal, hypothalamic hypophyseal portal system. This substance, which is called TRH, is eventually appearing into anterior pituitary. Now, the receptors for TRH are present on these cells. The cells which are supposed to synthesize and release TSH, right? Now, let me draw the cell in a magnified way and see exactly what is happening into this cell. If you draw this cell here, this is the cell, right? And here is TRH receptor. This is TRH receptor. Again, this TRH receptor is, yes, serpentine receptor, which is coupled with GQ intracellularly, and GQ, as you know, it will activate phospholipase C, and phospholipase C will act on the membrane of the cell, and from there it will release diacylglycerol and IP3 by breaking down phosphatidyl inositol diphosphate. This is what we have already learned in receptors, that when TRH, what is acting over here? The substance which act over here is TRH. This is released by group of neurons in paraventricular area, and these neurons secrete TRH into hypothalamic area. Hypothalamic hypophyseal system drain TRH to the anterior pituitary. There, this TRH will act on special serpentine receptors present on TSH secreting cells. When TRH bind with these receptors stimulate through GQ phospholipase C, which act on the phosphatidyl inositol diphosphate on the membrane and release intracellularly diacylglycerol and IP3. These second messengers these second messenger, when they increase in the cell, as you know, they will activate what? They will lead to stimulation of protein kinase C, and right, and especially IP3 will act on endoplasmic reticulum to release calcium, and calcium will also act on protein kinase C. So in a short, what we can say, that TRH actually through the G protein GQ activate phospholipase C, and that stimulate increase the diacylglycerol and IP3 levels in the cell, and that eventually increases intracellular calcium level. 3 caps. So we were talking about that TRH is stimulating the receptors on thyroid pituitary cells, right? And these serpentine receptors stimulate the GQ phospholipase C system. We degrade the phosphatidyl inositol diphosphate into IP3 and diacylglycerol. IP3, you know, it has action on the endoplasmic reticulum, and once IP3, that is inositol triphosphate, act on endoplasmic reticulum, it releases calcium. Intracellular calcium level goes up, at the same time intracellular diacylglycerol level goes up. When these two substances level go up, they activate different types of protein kinases, and protein kinases through the series of phosphorylation of target proteins lead to increased synthetic functions of the cells and lead to increased secretory functions of the cell. And as you know, the cell is synthesizing TSH as well as releasing TSH. So let's go back this cell over here. So we can say in short, when TRH is coming from the hypothalamus through the hypothalamic portal system, hypothalamic hypophyseal portal system, it act on the receptors, and when it act on these receptors, that will stimulate these cells, and these cells will release TSH. These cells will be releasing, yes, TSH, thyroid stimulating hormone. Now our next point is to understand how TSH work on thyroid stimulating hormone. Suppose here is your circulatory system, and we have to see how TSH is going to work on thyroid stimulating hormone. For this purpose, we will first draw here a thyroid gland, right? Let's suppose that here there is a thyroid gland. As you know, in thyroid gland, the functional unit to produce T3 T4 are follicles, and follicles are made of cuboidal cells, right? And these follicular cells are supposed to synthesize the TCT4 and release the TCT4. We'll remove one cell out of this follicle and we'll see how TSH work on that, right? So if we remove this cell we remove and put it over here. I will draw the cell like that, right? This is a follicular cell, right? Now, how this follicular cell particularly works, right? And how, by action of, let's suppose here is this follicular cell. So how TSH work on that? Again, there's a good news that as there were serpentine receptors present on this pituitary cells, in the same way the serpentine receptors which are present on the follicular cells. But these serpentine receptors are not meant for TRH, they are meant for TSH. So what really happens, this TSH is going over here, and this TSH is working TSH. This TSH is stimulating this particular serpentine receptor present on the basal membrane of the follicular cells. When TSH work over here, this receptor is activated, of course, and when this receptor is activated here, this unique type of serpentine receptor is coupled intracellularly with the G stimulatory proteins. And of course, all of you know, when G stimulatory is stimulated, This will lead to stimulation of adenylyl cyclase, and adenylyl cyclase, once it is stimulated under the effect of G stimulatory, right, what adenylyl cyclase is going to do? This is going to convert, yes please, ATP into, yes, ATP into cyclic AMP. So intracellular levels of cyclic AMP will become high, and this intracellular level of cyclic AMP will stimulate protein kinase A. Now this protein kinase A will lead to series of phosphorylation of many target enzymes and many target transporting proteins and many transcriptional factors. Again, let me tell you, TSH came on the follicular cells through the blood, right, on the basal membrane. There it bound with the TSH receptor, which was a serpentine receptor, which was intracellularly coupled with G stimulatory, and G stimulatory led to the activation of adenylyl cyclase, which broke down ATP into cyclic AMP. Here cyclic AMP is the second messenger system, and when intracellular cyclic AMP levels are high, the cyclic AMP will stimulate protein kinase A. And when protein kinase A is activated, protein kinase A will lead to phosphorylation of many proteins. For example, protein kinase A may lead to phosphorylation and alteration of function of iodide trappers. So iodide pumps will work more and cell will be taking more iodine, right? In the same way, protein kinase A will stimulate here even the enzymes thyroid peroxidases, right? And this thyroid peroxidases can do multiple function. Again, protein kinase A will not only phosphorylate the proteins which are concerned with thyroid uptake, iodide uptake, they will also lead to phosphorylation of this enzyme. And if this enzyme is stimulated, what will happen? Iodide will convert into iodine more rapidly, and meanwhile this will also phosphorylate the proteins which are going to the nucleus. And if nucleus become more active, especially the gene for thyroglobulin synthesis, then the cell will be producing more thyroglobulin. And if it is producing more thyroglobulin, right, more thyroglobulin is present over here. So what really happens, that more iodine is available, then this is also going to lead to extra organification of iodine, also leading and accelerating the coupling reaction between T1 and T2 or T2 and T2. So there's more synthesis of T3 and T4, and then increased endocytosis. There's increased endocytosis and intracellular degradation, and eventually increased release of T3 T4. So what we can say in a nutshell, that when TSH bind with the serpentine receptor that stimulate lead to stimulation of G stimulatory proteins, eventually through the adenylyl cyclase, it increases intracellular levels of cyclic AMP. When cyclic AMP levels are increased, what really happens? Protein kinases are activated. Protein kinases will phosphorylate many proteins in the cell, and when protein kinases will phosphorylate many proteins in the cell, all the functions of the cell will be increased. All the functions of the cell will be increased. Right now, what are the function of the cell? One function of the cell, it should trap the iodides, that is increased. Then iodide should be oxidized to iodines, that is increased. Then iodine should be bound with the tyrosine residues in the thyroglobin, and synthesis of thyroglobin should also be increased. Then monoiodotyrosine and diiodotyrosine should fuse with each other and coupling reaction should increase. Then endocytosis should be accelerated. Then degradation of thyroglobin to T3 and T4 should be activated, and eventually excessive amount of T3 T4 should be released. These were the early effects of TSH. These are the early effects of TSH, which start within few minutes to hours, right? So out of these, that all synthetic machinery and reabsorption machinery and secretory machinery, all of it is activated. What is the most important immediate effect of TSH? The most important immediate effect of rising TSH is, even though all effects are there, but the most important effect is the rapid endocytosis, so that preformed T3T4 can be rapidly released into circulation. Again, let me repeat it. In the early effect of TSH on the follicular cell is that in the follicular cell, TSH is basically what it is doing. It is stimulating the follicular cell. They are stimulating every aspect of the follicular cell related with the synthesis and related with the storage and related with the reabsorption and secretion of T3 T4, right? These are the early effects. Out of the early effects, the most important effect which is coming over there is, the most important early effect is, yes, that TSH lead to very rapid endocytotic process by the apical membrane of the follicular cell, so that colloid can be rapidly endocytosed, and then thyroglobulin should be degraded and T3T4 should be rapidly released into circulation. Is that right? Then the late effects of TSH. What is really the concept of late effect of TSH? Let me tell you. TSH has a short half-life. Half-life of TSH is only 1 hour. Half-life of TSH in circulation is normally only 1 hour. That is, of course, you must be knowing, only 60 minutes. These days in 1 hour there are 60 minutes, right? Now, what really happens? That TSH usually lasts in circulation only for 1 hour half-life. So within few hours it disappears. But if due to some reason, if TSH is present in circulation for longer time in excess amount, or there is some abnormal substance which is stimulating TSH receptor pathologically, right? Again, let me repeat it. Due to any reason, if TSH receptors are overstimulated for longer time, then long-term effects will start. Why they should be stimulated longer time, longer duration? Number one, maybe TSH is present in the blood in higher concentration and for longer time, or there are some antibodies, autoantibodies present in our body in some disease like Graves' disease. There are some autoantibodies which are formed in our body which bind with the TSH receptor and overstimulate. If those antibodies in place of TSH bind here and stimulate TSH receptor very powerfully, right? And you know, these antibodies keep on stimulating it for 12 hours, right? For very long time. Previously these antibodies were also called long acting thyroid stimulating substances, but now they are called TSI, thyroid stimulating immunoglobulin. Right. So my very dear doctors, we have already discussed what are the short term actions of the TSH receptor stimulation. And we were discussing, if there's long-term continuous stimulation of the TSH receptors, either by the normally naturally present TSH high concentration for long time, or by stimulation through the autoantibodies, as it happens in the Graves' disease. That in Graves' disease, thyroid stimulating immunoglobulins overstimulate the receptor system, right? What is real result? Short-term result, I will already know that there is increased synthetic and secretory activity. In the long term, what really happens is this long-term stimulation, then special, you can say, transcriptional factors are phosphorylated, and these transcriptional factors, when they are phosphorylated, they go to the nucleus and cell undergo hypertrophy and hyperplastic changes, right? Again, in case of prolonged excessive stimulation of TSH receptor system, there will be stimulation of genomic software, and cell will go in growth and proliferation stage, right? And what will be the real changes in the cells? Let's see in this diagram. Let's suppose this is the normal follicular cells and normal follicle. You know, normal follicular cells are cuboidal cells, right? Now what really happens? If there is overstimulation for longer time, these cells number will increase and they will become columnar. Now you see what has happened with this follicle, that due to excessive stimulation by the TSH system, the follicular cells number have increased, and rather than cuboidal, now they are, yes, now they are columnar. Not only cells number increase, not only their size increase, not only their length increase, but because cells become overcrowded and there may be infolding of the, you can say, unfolding of the groups of cells, right, towards the lumen, and lumen may not be a regular circle as it is normally seen in the normal. This is a colloid in a normal lumen of follicle, right? And here lumen has infolding, and due to that reason it is not in the normal shape. But if there is further excessive stimulation, right, what really happens, that then this, even not only the cell number increase, not only the length of cell increase, not only secretory activity of the cell increase, not only cells become crowded, but uh, number of follicles also increase. Number of follicles also increase, right? So out of one follicle, maybe you end up with multiple follicles. When this type of process is going on, you can understand it very clearly that total follicular mass in the thyroid gland will increase and thyroid gland size will be increased. And we say whenever there is thyroid gland size and weight more than normal, we say there is goitrogenic change, right, or goiter formation, right? So this is how we can depict in a diagrammatically when there is excessive and prolonged action of TSH on the follicular system. Is that right? Again, let me recap. Short-term overstimulation of TSH lead to increased synthetic and secretory activity of follicular cells. Long-term TSH receptor system stimulation leads to not only increased synthetic and secretory activity, but in addition to that, it leads to cellular growth and hypertrophy of cells and crowding of the cells, and you can say cells change their shape from the cuboidal to columnar, and per follicle number of cells increase, and eventually number of follicle in a given thyroid gland also increase, right? Once we have learned all this thing, now we come to another aspect, that how normally TSH secretion is regulated, right? Let's suppose if we talk about the normal cell from the beginning, that when this is hypothalamus paraventricular neurons release TRH, which are going through hypothalamic hypophyseal system, they are stimulating the cells in the interior pituitary, which are releasing TSH. TSH work on the follicular cells, that increases its all synthetic and secretory activity, and T3 T4 will come into circulation. Now already you know that T3 and T4, when they are released in circulation, T3 is released in smaller amount and T3 is released in smaller amount, maybe only 10%, and T4 is released in larger amount. Of course, most of this is not present in circulation as free. As you know, there are thyroid binding globulins, right? So T4 mostly, and along with it some T3 also bind with thyroid binding globulins. Thyroid binding globulins, right? Now they are carrying more than 90% of T3 and T4 is bound with the thyroid binding globulin, and only less than 1% of T3 and T4 is free. But you have to remember that T3 is the active form which acts rapidly and more powerfully on the target cells. As you know, T3 poorly bind with the plasma proteins but strongly bind with the target receptors in the nucleus, right? Opposite to that, T4 more strongly bind with plasma proteins and poorly bind with the target receptors. Due to these reasons, T4 is biologically less active and T3 is biologically more active. As you know already, that T4, once it is free and it will diffuse to the tissues, most of the T4, very little T4 directly act, most of the T4 will be acted upon by 5' deiodinase enzyme, which will remove one iodine from the T4 and convert it into T3 so that it can work more avidly and more powerfully on the target cells. It means T4 is basically a pro-hormone or storage form of the hormone in circulation. Now, this thing has another implication. Now, you have to be very careful to understand this point. Let me repeat. In the circulation, total T4 is far more than total T3, right? So feedback mechanism is mainly dependent on circulatory T4 level, not on T3 levels, right? Now, how it really operates. T4 levels. T4 has a negative feedback mechanism, and T4 can enter into, yes, enter into which cell? Thyroid cells. T4 can enter into thyroid cells, and there it could get converted into T3. Now all the time T4 which is present in circulation, a very small amount of that is taken up by the pituitary cells. What are these cells? Pituitary cells. And this T4 which is taken up by the pituitary cell, within the pituitary cell is converted into T3, and this T3, right, down regulates the receptor for TRH. Is that right? This T3 down regulates the receptors for TRH. Or we can say this is a circulatory T4 which enter into the pituitary cell, convert into T3 and down regulate the receptors for TRH. It means as your circulatory level of T4 goes more and more up, as in your circulation T4 levels go up, it means the number of receptors and sensitivities of receptors on the pituitary cell keep on decreasing. That is why we call it negative feedback mechanism. Now, on the pituitary cell, when TRH receptor have gone down in their number and their sensitivity, it means further release of TSH is reduced. So, how we can say? Let's suppose if in circulation your TRH T4 level is less, then TSH producing cells become more sensitive to the action of TRH. And if in circulation T4 level is high, then pituitary cells, TSH releasing cells become less sensitive to TRH. So in this way, by circulating T4 levels regulate the sensitivity of TSH producing cells of the anterior pituitary towards the TRH, right? Is it clear? So what do we learn now from this? We learned it that in a person, if hypothalamic pituitary thyroid axis is working normally, if in a person this system is working normally, and due to any reason, if T4 is chronically low, TSH will become high, plasma level of TSH will go up. Or in another person, if T4 levels are chronically high, then eventually TSH level will go down. Is that clear? Now another factor which is important in regulating the TRH and TSH T3 T4 system is temperature, cold. It has been seen that if lower animals are exposed to cold for longer time, the synthesis of T3 T4 increases. How exactly it happens? Now it is believed that actually in the hypothalamus there are certain areas which are temperature sensitive, right? The neurons in the hypothalamus which are temperature sensitive, and these neurons also play a role in regulation of TRH. For example, if your body temperature is less, right, you need more heat in the body, you need more basal metabolic rate. So what really happens, the temperature sensitive, rather I must say cold sensitive neurons stimulate those neurons which are supposed to secrete TRH. So what really happens, that if your body temperature for core body temperature is low for longer time, then hypothalamus is releasing more TRH, of course, then anterior pituitary must be releasing more TSH, and of course then synthetic and secretory activity of follicular cells will increase, and body will be provided with the more T3 T4, so that basal metabolic rate should go up and heat should be produced. Then another interesting finding is that if you are under emotional stress, or you are stressful, like severe worry or anxiety, your TSH level changes. What is the reason? Now they believe that when you are under severe stress or there is a lot of anxiety, what really happens in your body? There's a lot of activity of sympathetic nervous system. And you know, sympathetic nervous system, epinephrine and norepinephrine, they are calorogenic, right? They produce heat in the body, and that heat inhibits the cold center here and reduces the production of TRH, which eventually lead to reduced production of TSH. So we can say that hypothalamic areas can regulate the production of TRH and eventually TSH and T3 T4 by under the influence of temperature as well as emotions, right? After this, there's one very interesting finding I would like to narrate, and that is low T3 syndrome. Low T3 syndrome. What is low T3 syndrome? We can logically talk about the syndrome, right? Low T3 syndrome. You see, if you are suffering with any catabolic disease, if you are suffering with any catabolic condition, for example, there you are under stressful catabolic state, stressful catabolic state, yeah, for example, you are starving, prolonged starvation, prolonged starvation, or there is surgery, extensive surgery, or there's extensive trauma, or there's high grade fevers, right? Or there is any type of serious illness which is leading to catabolism of your body. Under these circumstances, body will love to reduce the level of T3 so that T3 mediated catabolism should not occur. How the body is doing it? It's very simple, that whenever there are stressful circumstances, right, they inhibit the enzyme in the peripheral tissue, the enzyme which is called, yes, 5 prime mono deiodinase. You know, this enzyme is present in your peripheral tissue, and duty of this enzyme is to convert the T4 into active T3. Is that right? Now, if this enzyme is inhibited, what really happens that in these patients, level of T3 will become low because this enzyme is unable to convert T4 into T3. And when level of T4 becomes less, what is the result? Sorry. Level of active T3 is less. Please correct it. When this enzyme is not working, T4 cannot be converted into active T3, so level of active T3 is less. But the point which you have to remember in these patients, but levels of T4 are normal as well as levels of TSH are normal, because TSH is sensitive to the level of T4, not to T3, right? So this is a person who is having a stressful catabolic state, and his peripheral enzymes are not converting T4 into active T3 efficiently. So in his body, level of active T3 is going down, so T3 mediated catabolism is reduced, right? But what really happens, the T4 levels remain normal in the blood, and you know TSH is sensitive to the T4 level, so naturally when T4 levels remain normal, TSH levels also remain normal. Is it clear to everyone? Now, in a nutshell, let's talk about what are the factors which stimulate the release of TCT4, and what are the factors which inhibit the release of T3 T4. We'll be talking about what are the factors which stimulate the release of factors which increase T3 T4 levels in the blood, and of course factors which decrease T3 T4 levels. Now, it's very easy to understand the factors which basically increase T3T4 level. Of course, please tell me TSH. TSH, when its level rises in your blood, it acts on the thyroid cells, and you know it produces more T3T4. Number two, thyroid stimulating immunoglobulins. As in Graves' disease, they also increase the production of T3 and T4. Then total level of T3 and T4 may be increased when when your liver is producing excessive amount of thyroid binding globulins. Increase levels of thyroid binding globulins, right? Whatever the cause. If in your body total level of TBG increases, then total T3 T4 also increases. But remember that free T3 T4 may not be significantly changed. What are the conditions from the liver which can lead to excessive amount of TBG release? Number one is pregnancy. In pregnancy, estrogen progesterone stimulate the hepatocyte to secrete more TBG. Or the females who are on oral pills, right? In their body also, liver has increased synthetic capability of thyroid binding globulin, that translates into total rise in total level of T3T4. Opposite to that, when we talk about what are the factors which inhibit the T3T4, of course, that must be low TSH. Low TSH will reduce T3T4. Then we can talk about low iodides. Sometimes dietary iodide is low. Not enough iodine is provided to the thyroid gland to synthesize T3T4. Or, very interestingly, very high level of iodides. As I told you in previous part of the lecture, that very high level of iodides can also inhibit the iodides uptake. This, and the special name for this condition was, yes, this was Wolf Chaikof effect. Then we can talk about certain antithyroid agents which can reduce T3T4 level. Right? We have already discussed those substances in detail in previous lectures. For example, thiocyanates, perchlorates, perchlorates. These two act on iodide pump and inhibit that. Then, of course, we should not forget our very important friend thiopropyl uracil and its related compounds like methimazole or like carbimazole. All these can inhibit thyroid peroxidase and inhibit multiple steps in T3T4 synthesis. Then, of course, there's an enzyme present within the thyroid cells which is called thyroid deiodinase. Deiodinase. And this enzyme was concerned with recycling of iodine from monoiodotyrosine and diiodotyrosine. Right? If this enzyme is absent in the thyroid cell, then iodine which is attached with the monoiodotyrosine and diiodotyrosine that cannot be recycled, and symptoms of low T3T4 like hypothyroid thyroid symptoms will present. Then another interesting situation in which T3T4 total levels may go down is low plasma proteins. As you know that in high levels of thyroid binding TBGs, in high TBGs, right, T3T4 levels go up. If low TBGs, naturally total T3T4 level will go down. For example, in cirrhosis, patient who have cirrhosis where liver has reduced its synthetic functions and serum level of thyroid binding globulins will go down, and of course then total T3T4 level will also go down. Other situation which is there is loss of proteins out of the body. For example, nephrotic syndrome. Nephrotic syndrome, you know that through the renal system, there is injury to glomeruli, and glomerular system is losing unduly large amount of albumins and even sometimes globulins in urine. And if there's severe glomerulonephritis, what really happens that not only serum albumin levels go down, but also serum globulins level go down, and total T3T4 levels become less. So that's all about regulation of T3T4 secretion. Yes, now my dear students, we are going to learn how thyroid hormone works, especially how the T3 works on our tissues, right? So already you know that T3 is produced by the follicles which are present in the, yes, thyroid gland. And from here T3 and T4, normally T3 is produced in small amount and normally T4 is produced larger amount, and both of them are secreted by the thyroid gland into general circulation. And already you know this T3 and T4 is carried in general circulation in association with the proteins, plasma proteins, especially thyroid binding globulins, right? And again you know that most of the T3 and T4 they are binding with the TBG, and less than 1% is free T3 or T4, right? Then and now we have to see that this free T3T4 is passing through your circulation all over the body, and how it work on the target tissue. Remember, T3T4 or thyroid hormones work on almost every cell in the body, right? And they have a general mechanism of their function, how they're working. Let's see it. Suppose here is the target cell. This is a particular cell, and we are going to see how T3 and T4 work on this particular cell. Now, first thing which is there is that you must know that protein bound T3 T4 cannot act on the cell. It's the free T3 or T4 which is biologically active. So what really happens? That T3 is coming there to work, and if T4 is coming, T4 needs to be converted into T3 in most of the cases. The reason being that T4 has strong affinity for the plasma protein and carrier proteins, but T4 has a weak affinity to bind with the ultimate receptors, right? Due to this reason, there are special type of enzymes, and these special type of enzymes which can convert, you can say, T4 into T3 by removing one iodine. These enzymes are called 5' yes deiodinase, monodeiodinase enzymes, right? So what is the end result? The T3 itself, or you can say T4, after one iodine is removed while it is converted into T3, right? This is the ultimate final product which mainly works. There's very little T4 which directly work on the receptors. Now we have to see how this T3 exactly work on the receptors. The target receptor for this T3 is inside the cell, inside the nucleus. You know, free T3 is a very small molecule. It is a derivative of single amino acid, and it can easily enter into cell, right? Because this substance can easily enter into cell, its receptors should be present within the cell. Actually, its receptors are present within the nucleus. Now let's suppose I draw a nucleus here, right? And let's suppose this is the nucleus of this particular cell, and we have to see that how this T3 is going to work over there, right? Here is T3 entering into cytosol, and eventually it enters into, yes, nucleus. We can represent T3 by, let's suppose, yeah, this is your beautiful T3, right? This is T3. Now we have to see, once T3 is in the nucleus, how it is going to work. You know, in the nucleus there is genetic program, right? There are genes, the genetic programs, and this T3 has to run many genetic softwares or genetic programs or genes and activate them. Let's suppose that here is double-stranded DNA, right? And a part of this double-stranded DNA is acting as gene. Let's suppose from this point up to this point, the nucleotide which are present, they are acting as a specific gene which is capable of making a unique type of messenger RNA. But you know, genes can be activated by many factors. And many of the genes which are present, they can be activated with the help of T3. Now, how this T3 can activate this particular gene? Actually, it has to first bind to its own receptors, the receptors which are nuclear receptor, right? The receptors which is present over there, right, where T3 can bind, this is T3 receptor, and this is the domain where who can bind? T3. And this is the receptor, but its its active domain is normally not active. Let's suppose this receptor has this specific domain with which T3 will bind, this specific domain with which T3 will bind. So you can say this this is ligand binding domain, and this is the domain which has to bind with a specific nucleotide sequence here. With a specific, within the gene, it has to bind with a specific nucleotide sequence, and this is called hormone responsive DNA elements. Hormone responsive DNA sequences. So what really happens that here I've made a specific receptor for the T3, and this receptor is inactive. This receptor is right now inactive. Right now, how the T3 will activate again? This receptor has one point where T3 will bind, then it has another point, and this point is supposed to bind with the hormone. This point is supposed to bind with the hormone responsive element. It means this is effector domain. And there's one more point. I will tell you how this domain works. So domain number one is, this is domain number one, which is ligand binding domain. This is domain number two. I will tell you its function later. And this is the true domain number three, which is going to produce its effect. So domain number one is for the ligand binding domain or T3 binding domain. And domain number three is effector domain. Right? Now, what really happens? Actually, when T3 binds with this domain, again, what was this domain? Now, here T3 has bound. It's working very hard. This is T3. This is binding with this, you can say, receptor binding, T3 binding domain of the receptor. Now, what T3 is doing? It is pushing the receptor, and as soon as this binds with this domain, its effector domain is activated. Which domain is activated? Effector domain. Right? This effector domain is activated, and now this effector domain can engage with the hormone responsive element. Is that right? Now, as you know, everyone who is doing a great job needs helpers. In the same way, this T3 also needs its friends, right? Which will help it to further enhance the action of the receptor on the DNA, right? So there's one more domain here. I told you there was one more domain here, and here is a very small friend of T3, and this also helps it to bind and help into in that particular situation. I think a bit stylish, right? So it is also working hard to activate the system. This is also working hard to activate the system. Now, this one is your, yes, what was this? T3. This is T3. And what is this one? There is a special name for it. Yeah. What is the name of this? This is actually itself a receptor for retinoic acid. So it is called, this particular molecule is called RXR. It is not a name of a car. RXR means retinoic acid X receptor. So what we can say that T3 bind with its receptor, right? And RXR receptor act as a co-stimulator. T3 is the primary stimulator of the receptor. RXR are co-stimulator, and once this receptor is properly stimulated, then this receptors, which domain? Yes, please. Effector domain. This will bind with hormone responsive element in the DNA. When this hormone responsive element in the DNA is acted upon by this receptor, of course I should write, this is T3 receptor, right? When hormone responsive domain is stimulated by the active receptor, it will alter the transcription of gene. In most of the cases, in most of the genes, it will increase the rate of transcription of the gene. It means that gene will start the process of transcription, and from the DNA, gene will start making RNA. So there will be formation of, yes, what? New RNA. From this DNA, there will be formation of what thing? This is pre-messenger RNA, and after some processing and changes, this messenger RNA will appear into cytosol. And when this messenger RNA will appear into cytosol, as you know that messenger RNA will be acted upon by many, yes. What are these? ribosomes, and when they are acted upon by many ribosomes, these ribosomes start the function of translation. And when these ribosomes start translating the language of the messenger RNA into amino acid sequence, so there will be formation of, what is this? Yes, what is it? Formation of new peptides and proteins, right? So in this way, what T3 has done, again let's recap. The basic function of T3 is that T3 can enter into a cell, right? It can cross the cell membrane easily, it can cross the nuclear membrane easily, and within the nucleus, T3 will bind with its T3 receptors, and it will activate those receptors, and it will be having a help from another factor, which is RXR, or retinoic X receptor, right? Then this complex will have an active effective domain which will stimulate the hormone responsive sequence in the DNA of a gene, and this gene will be activated, and this gene will start the transcriptional process. So you can say this particular receptor could activate the transcription of a particular gene. Due to this reason, T3 receptors are also called unique type of transcription factors. What are T3 receptors? T3 receptors are T3 or T4 responsive transcription factor. Again, let me repeat it. T3 binds strongly with these receptors and T4 binds weakly with these receptors. Due to this reason, T3 is 4 to 5 times more potent than the T4 action. Right? Major function of T3 is that it activates the transcriptions of many genes. It is not activating one gene. In this model, I have shown only one gene, but actually when T3 molecules enter in any given cell, they will activate hundreds of the genes. And if the hundreds of the genes are activated, it means hundreds of new messenger RNA are made, and hundreds of new proteins are made in every cell after the action of thyroid hormones, right? So we can say what thyroid hormones are doing. Thyroid hormone enter into cell, they bind with special type of receptors which are transcriptional factors. Those transcriptional factors bind with multiple hormone responsive element, and they alter the genomic expression of the cell, and hundreds of the genes are activated. And when hundreds of the genes are activated, hundreds of the new messenger RNAs are made, hundreds of the new proteins are made in the cell. And these new proteins which are made in the cells, what will happen? These new proteins, right? Of course, some of them will be functional proteins. Some of them will be, yes, structural proteins, and again some of them will be regulatory proteins. Now, these functional proteins, right, these functional proteins which are made in the cells, right, they will increase the metabolism of cell, increase the, yes, metabolism of cell, because many of these functional proteins are just enzymes. What are these? Enzymes. So it means that T3 can activate the receptors which can activate the genes for the enzymes, and many new enzymes appear in the cell and cell metabolism alter. Just to say it shortly, capability of cell to handle the glucose is more. Capability of cell to utilize oxygen is more. Oxidative phosphorylation reactions and enzymes which are involved in that are more active. And so many other enzymes are increased. In the same way, when we talk about the genes which are concerned with the structural aspect of the body, if their transcription is altered and more structural proteins are made, these structural proteins will play a role in growth and different maturity. Growth, right? And this growth may be mental maturity, or it may even involve physical maturity. For example, look here very carefully that children who are hypothyroid, they don't grow well, right? The physical growth is hampered. Even their mental maturity is hampered. They remain mentally retarded. Am I clear? Then regulatory proteins. Regulatory proteins may be many types of proteins which are regulating different functions. For example, A very important regulatory protein is, yes please, regulatory protein like different transporter proteins. For example, sodium potassium ATPase, sodium potassium ATPase, right? Actually, after the action of TCT4 in every cell, sodium potassium ATPases concentration is increased in their membrane. So every cell become loaded with more and more sodium potassium ATPases, and you know this is one of the major point where a lot of ATP is utilized. It means a lot of oxygen is utilized, a lot of heat is produced, and basal metabolic rate will go up. Is that right? Even another example of regulatory protein is, for example, on the surface of hepatocyte, there is increased expression of LDL receptors. Of course, the capability of liver cell to take up the cholesterol is increased. So what I really want to, in a nutshell, by this explanation, what I really wanted to tell you, that T3 or thyroid hormones enter into every cell in the body. They enter into cytosol, then they cross the nuclear membrane, they enter into nucleus, they bind with their target receptor, they take some assistance for retinoic acid receptors, right? And then their receptors which are transcriptional factor, they will alter the transcription of certain hundreds of the genes, and hundreds of genes will start transcribing. They will produce hundreds of new messenger RNAs, and these messenger RNAs will be eventually translated within a few hours into few minutes to few hours. These new messenger RNAs will be translated into new proteins, and then cell will have a lot of new functional proteins and enzymes, structural proteins, and alteration in its growth pattern, and it may have a lot of regulatory proteins and different type of transporting proteins. So this is, in generally speaking, how the TCT4 works. Is that right? Now we'll be talking about what are the major effects of, you can say, T3 T4 on your body, right? If you really want to know it as brief as possible, we can say there are three basic effects. Number one, that thyroid hormones increase basal metabolic rate. Number two, they play a role in physical and mental maturation, physical and mental growth and maturation. And number three, they maintain responsiveness of body tissues to adrenergic drive. Responsiveness of body tissues to adrenergic drive, because T3T4 increase the expression of adrenergic receptors, especially beta receptors. Now, first question we have to answer, that how T3T4 increase the BMR, and how much they increase the BMR, and in which tissues it increase the BMR, basal metabolic rate. Answer is that if you increase the dose of T3T4 too much, the increase in basal metabolic rate may be 60 to 100% increase in BMR. And if you take the T3T4 level dangerously down, right? For example, this is a normal BMR, you can double it. Or if you remove the T3T4 out of your body, BMR will become dangerously low, and it may fall to its normal 50% of the normal value. So it means that if you bring the T3T4 level at zero, right, BMR, basal metabolic rate, will be there, but it will be less than about 50% of the normal value. And if you increase the T3T4 in your body, as it happens sometimes in thyrotoxicosis, right, thyrotoxic states or hyperthyroidism, then basal metabolic rate is increased. In our further discussion, we have to discuss into detail that how the basal metabolic rate in the body is increased by this hormone, and how it has increased basal metabolic rate has different effects on different systems on our body. Secondly, we have to talk about that this hormone also play a major role in physical and mental growth and maturation. As you know, the children who are born with hypothyroid states, they are physically not growing properly, they're short-heighted, and at the top, they are mentally not mature. They have mental retardation, especially three areas in the brain which are very sensitive to the presence of T3 T4. That is cerebral cortex. When cerebral cortex does not develop properly, a child may have mental retardation. Secondly, basal ganglia don't develop properly, so child may have rigidity. Is that right? In the same way, then another important aspect is that these hormones maintain the responsiveness of the tissues to the adrenergic drive. I've already told you that if there's good levels of T3T4, they will maintain the good expression of adrenergic receptors, especially beta receptors, for example, beta 1 receptor on the heart. Is that clear? Now, we'll discuss all these things into detail. First of all, we'll talk about that how it increases the BMR in our body. Remember, there are some tissues in our body in which T3 T4 do not increase the BMR directly. Again, T3 T4 increase basal metabolic rate in almost every tissue, but there are few exceptions. Those tissues, right, on those tissues T3 T4 does not directly increase the BMR. So let's keep the exceptions on the side. The exceptions are your central nervous system, then there is retina, CNS, retina, lymph nodes, and of course your dear gonads. Gonads, lymph nodes, spleen, retina, and central nervous system, right? T3 T4 does have effect on these tissues indirectly, but T3 T4 does not directly increase the basal metabolic rate of central nervous system or gonads or retina or spleen or lymph node. If you really cannot remember all of them, at least remember that T3 T4 does not increase the basal metabolic rate in CNS and does not increase the basal metabolic rate in gonads. Now it's up to you that you should have to think over what is common in your central nervous system and in your gonad that T3 has really sensed and decided not to increase the BMR. But remaining tissues in the body, it increases the BMR. Now let's come to the question that how T3 T4 increases the BMR, how thyroid hormone increases the BMR. Let's recap a little bit and then we'll go for that. As you know already, that basically hypothalamus produces TRH, which act on the anterior pituitary cells, and from there it releases TSH, thyroid stimulating hormone. TSH will bind with TSH receptors on thyroid follicular cells, and the follicular cells will synthesize, store, and release extra amount of T3T4. T3T4 99% is bound with the plasma proteins, only less than 1% is free. This free amount of T3 can enter into cells. Even before entering into cell or within the cell, T4 can be deiodinated and converted into T3. Once T3 is inside the cell, as we discussed in previous session, that T3 will bind with its nuclear receptors along with retinoic acid receptors and stimulate hormone responsive elements in multiple genes, and that will lead to hundreds of new messenger RNA expression within the cell, and that will make hundreds of new proteins. Right now, these proteins and other changes in the cell will take the basal metabolic rate up, and generally functions of all systems in the body will go up. And remember, if T3 T4 level is very high, the systems will become so fast or so active that they don't work properly. But anyway, what are the real effects? I told you one of the protein which is produced is, yes, sodium potassium ATPases. And if a lot of sodium potassium ATPases are produced in your cells, remember normally every cell has sodium potassium ATPases, but if your T3 thyroxine level goes up or T3 T4 level goes up, then concentration of sodium potassium ATPases and the cell membranes increases. And in hypothyroid patient where T3 T4 level is less than normal, then in their cell membrane sodium potassium ATPases, you can say concentration decreases. And now one thing which you must be knowing, that sodium potassium ATPases of course use the ATP for their function, and what they are doing while they are using ATP, they are constantly pushing the sodium out of the cell they push 3 sodium out of the cell, and for every 3 sodium thrown out of the cell, they will bring just 2 potassium into the cell. Due to these sodium potassium ATPases, which are normally present in every cell accumulates potassium, and it's a bag of potassium. Every cell is very rich in intracellularly rich in potassium, and extracellularly every cell has a lot of sodium. Right now, this is due to these sodium potassium ATPases, and these sodium potassium ATPases use very abundantly lot of ATP, lot of energy, and they produce a lot of heat in the body. Now listen, if T3 T4 levels are going up, it means function and concentration of these transporters are more. So it means utilization of ATP is, yes, more, and it means need of oxygen is more by the cell, because to produce more and more ATP to give supply to these sodium potassium ATPases, you need more oxygen, you need more nutrients, you need more glucose, you need more fatty acids, and of course the real storehouse, real factory in the cell which produces ATP, which help in production of ATP, all of you know that. What is that? Mitochondria. So what really happens, that to provide enough energy, because energy demand on the cell is too much, you have to supply the cell with oxygen, you have to supply the cell with extra oxygen, extra glucose, extra fatty acid for beta oxidation, you have to supply a lot of nutrients. But of course, all these nutrients and oxygen will help to produce ATP through mitochondrial function. So what really happens? Mitochondrial size also increases. Look, as T3 T4 level goes up, mitochondrial size also increases. Not only size of the mitochondria increase, with excessive function of T3 T4, number of mitochondria also increases. So it means that for people who are having hyperthyroid state, they are having not only larger mitochondria in their cells, they are having more number of mitochondria also there. And of course, when size of the mitochondria increases, when the number of mitochondria increases, it means overall surface of mitochondrial membrane increases, and of course it means all the enzymes which are working in relation to mitochondria, oxidative phosphorylation enzymes, which are supposed to use the fuels in the body and produce a lot of ATP, those enzymes concentration also increases. In a nutshell, we can say T3T4 acts on every cell in the body, right? And especially other than the CNS, gonads, spleen, and lymph nodes, especially other than these tissues which I have mentioned, it increases basal metabolic rate. How it produces basal metabolic rate? So many ways. Some even author believe that increased T3 T4 increases the sodium influx in the channel. Sodium influx in the cells through increasing the channel. This increased sodium influx also activates the sodium potassium ATPases. Secondly, as I told you, that nuclear T3 T4 has to go to the nucleus. They have to activate the transcription of a lot of new genes, produce a lot of new proteins, and out of hundreds of the protein, one protein which is very important is sodium potassium ATPases. Other group of proteins are the enzymes, right? So sodium potassium ATPases are more numerous and they're more active. They are utilizing more energy, more ATP. To provide more ATP, we need more enzymes, and we need also more factories to produce energy. So mitochondria not only increase in their size, but they also increase in their number, right? So if next time someone asks you that how T3 T4 increases the basal metabolic rate, answer should be very simple. What should be the answer? T3 T4 through the acting action on the nucleus increase the transcription of many genes, many new proteins are produced, especially many metabolic enzymes, many transporter proteins, right? If you have to mention especially which transporter protein, at least don't forget to mention sodium potassium ATPases. And if someone asks which enzymes, of course so many enzymes, but at least don't forget to mention mitochondrial enzymes. Is that clear? Of course, all these things will increase the basal metabolic rate of the body. Is that clear? Right. So we were discussing about physiological actions of thyroid hormones, and about the physiological actions of thyroid hormones actions of thyroid hormone. Few points we have already discussed in detail. For example, we have discussed that thyroid hormones lead to increased transcription of large number of genes, increased transcription of many genes. This is what we have discussed already. Second point which we have already discussed is that thyroid hormones increase cellular metabolic activity. They lead to increase cellular metabolic activity, right? In that regard, we specially talked about increased number of sodium potassium ATPases and increased number and size of mitochondria. Third point which we have to talk about the functions of thyroid is that the role of thyroid hormones on growth and maturation, role of thyroid hormones on physical and mental growth and maturation. And in the end, we will talk about in detail some other aspect. So let's concentrate physical and mental growth and the role of thyroid hormone. Let me tell you some interesting thing. You know about tadpoles. Yes, what are tadpoles? Baby frogs. Is that right? Now look, scientists have done very interesting experiment to prove that thyroid hormones have very powerful action for growth and differentiation and maturation for physical and mental. For example, if normally what happens if T3 T4 concentration is normal, tadpole gradually develop into mature frogs, right? Now, if you deprive the tadpoles from T3 T4, if there's no T3 T4, you don't provide any T3 T4 to the tadpole, you'll be surprised to know something. Tadpoles will always remain tadpoles and they will never develop into frogs. They will never develop into frogs. This is one very strong proof that without T3 T4, physical and mental growth is not there. Is that right? Neuronal system does not mature. Second interesting thing, if tadpoles are provided with excessive amount of T3 and T4, they very rapidly mature into frogs, but mini frogs. Because if they give the normal look again, here is your friend tadpole, and with normal, let's suppose, with the normal T3 T4, this tadpole will develop into normal frog. I don't know how to draw a frog. You just imagine this is frog, right? If there is no T3 T4, what happens? No T3 T4, what will happen? Tadpole will always remain tadpole until they die. And if you give excessive amount of T3 T4, tadpole will convert into frogs, but these are mini frogs. These are mini frogs. So what does it mean? That for the normal growth and maturation, you need normal concentration of thyroid hormones. Subnormal concentrations are not good. Even above normal concentrations are also not good. Now, like tadpoles, these facts are also true about human beings. The children which are born with hypothyroidism, children which are born with hypothyroidism or hypothyroid state, the problem in physical growth as well as mental growth. As I told you previously, these children will grow very gradually and their total height achieved will be less than normal, but soft tissue grow relatively better. Due to that reason, these hypothyroid children become short heighted but obese, right? Heavyweight, and mentally they remain retarded. And sometimes soft tissue so much like tongue cannot be accommodated into their mouth like this. Is that right? We call these baby children cretins. What we call them? They're suffering with cretinism. Cretinism, hypothyroid babies. When they grow up, they're suffering with cretinism. They're having mental retardation. Now the question is that why do they have mental retardation? The reason being, when T3T4 is not there, neuronal circuits don't develop properly. Number one, in central nervous system, what are the troubles? Basic problem is synapses don't develop properly. Synapses in between the neurons are abnormal. Number two, dendrites don't develop. If neurons are not developing dendrites, can they develop proper connections with each other? Number three, neurons don't develop proper myelination problems. Myelination. So the myelination problem, the dendritic growth problem, and synapses are abnormal. All these things lead to abnormal central nervous system function when T3T4 is low, right? Especially cerebral cortex does not develop well, and baby will suffer with mental retardation. Basal ganglia do not develop very well, so baby will suffer with rigidity. Baby will suffer with rigidity. In the same way, even physical growth is not well, bones are not growing properly, they are short heighted, and chronological age and bone age are different. Normally, from the bone x-rays and bone age should be related with the chronological age. Here, chronological age of the baby may be more, but bone age of the baby will be less because bones are immature and not developing properly. Is that right? Okay. But now we imagine the people who are hyperthyroid. What do you think, doctor? If a child is from the very early life is given extra thyroxine, what will happen to his height? He will be taller. This is something which is wrong, rather partly right and partly wrong. Let me tell you how. Actually, if there are two babies, this is the normal baby and this is the baby with hyperthyroid state. Initially, hyperthyroid state baby, both babies are growing, but hyperthyroid state baby will grow at a faster pace. Due to that reason, during the development, initial phases of development, it will look as if the eventual height of the hyperthyroid baby will be more, but it is not true. Why? Because hyperthyroid baby initially grows rapidly but closes its epiphysis in the bone earlier. But normal baby keep on developing gradually and eventually it becomes taller. So what really happens? The children who are hyperthyroid, even though initially bone grows very rapidly, but growth arrests very early due to closure of the epiphyseal plates. Due to this reason, again hyperthyroid babies are also what? Dwarf. And hypothyroid babies are also dwarf. But there's a difference. That hypothyroid babies will be mentally retarded and they will be short heighted and with heavy weight. But hyperthyroid baby will be mentally anxious, right? And the hyperthyroid baby will be underweight and again they will be also dwarf, right? So by all this discussion, what I really wanted to tell you that T3T4 has a major role in development of, you can say, for physical and mental maturity. Another interesting situation is that T3T4 work has an anabolic action in its normal concentration along with growth hormone and other growth factors. Rather, it's important to remember that T3T4 help in increase synthesis of growth hormone as well as secretion of growth hormone. Am I clear to everyone? Fine. Right. So we were talking about the physiological actions of T3T4. We have already discussed that one function of the T3T4 is that it should increase the transcription of many genes so that lot of proteins are made and then its actions are executed. Then we have already discussed that thyroid hormones increase cellular metabolic activity, in which reference you must remember that there is increase ATPase, sodium potassium ATPase activity and increase size and number of mitochondria. Then we have already discussed that Third point is that thyroid hormones play a major role in physical and mental growth and maturity of young adults, right? And children development, right? After that, now we come to the last but very important aspect of thyroid hormone, that what are the thyroid hormone action on multiple systems and mechanisms in the body to coordinate, so that multiple mechanisms in our body are coordinated in such a fashion that BMR is maintained, growth is maintained, maturation is achieved, right? So now we'll talk about effects of the thyroid hormones on specific bodily mechanisms. Effects of the thyroid hormone on specific bodily mechanisms, right? So to explain this, I really need a master diagram for you, right? So I think I must rub this, and now I go to explain the make a master diagram. Okay, let's start. First we will make the general circulation and many systems in our body, and then we'll see how multiple systems in our body are influenced by action of T3 and T4, right? Okay, you must know, here is your right heart. Let me make a simple diagram, and here lies your lung. Is that right? And of course you should not forget that there are airways there, and this is air pockets, and let's suppose this is your pulmonary circulation. Here it takes oxygenated blood and then it drains to the left side of the heart, and from the left side of the heart blood is supplied to all over the body, and eventually this circulation will come back to, yes please, to which side? Right side of the heart, right? So you know, between the left heart and the right heart, here there is systemic circulation, right? And between the right heart and the left heart there is pulmonary circulation. Now we have to see that what really happens to our body functions under the influence of T3 T4, right? Now to understand this thing, first thing which we know already, that what is happening, that yes, let's suppose these are typical cells in the body, right? These cells represent all cells in the body except your central nervous system and gonads and spleen and lymph nodes and retina. These cells may be liver, it may be GIT, it may be any cell in the body. These general cells in the body, when they are acted upon by T3 and T4, you know, mitochondrial number and size increases, sodium potassium ATPases increases, lot of enzymes concentration and action increases, so basal metabolic rate goes up. We already know. It means that if we are talking about this general cells, so we say there is increased basal metabolic rate when there is a good supply of thyroid hormones. Now, question is that how the different part of the body will act to help this basal metabolic rate. First thing is that you have to supply the fuel. You know, when basal metabolic rate is more, it means you're producing less ATP or more ATP. It means all over the body more ATP is produced in all the cells, and more ATP is not only produced, more ATP is utilized as well, right? If body has a very good metabolism, high rate of metabolism, more ATP is being produced and utilized, it means we have to provide the raw material. If we have to provide the raw material, it means we have to provide the fuel, we have to provide the glucose, we have to provide the fatty acids, as well as we have to provide more oxygen, right? So how our bodily functions coordinate to provide more fuel and more oxygen, and how bodily functions may work in such a fashion to remove more waste products. Okay, let's start with carbohydrate metabolism first, right? You tell me that this person need more glucose in his body or less. So naturally what will happen, that his GIT is coordinated in such a way that under the influence of thyroid hormones, GIT will absorb more glucose. GIT will absorb more glucose, right? And to absorb more glucose, it means GIT functions need to be increased. Is that right? Yes. And if GIT functions need to be increased, it means GIT has to produce more secretions. So secretory activity of GAT has to increase, right? Absorptive capacity of GAT has to increase. I'm talking about normal range of T3 T4. Of course, in hyperthyroidism, these functions will become pathological, right? And of course, GAT motility should be good, right? And with that, of course, we should not forget it should maintain the appetite. If there's good appetite, there's good food intake. The diet should be good, right? So appetite is good. When T3 T4 is working well, your bodily metabolisms are good. You need more energy. Let's start with the carbohydrate, right? Carbohydrate number one, GIT, glucose absorption should be more. Not only glucose absorption should be more, my friend, you must know that liver, which is one of the very important regulator of carbohydrate metabolism, that is also playing its role, right? When T3 T4 is more, right, what will happen in the liver, right? There will be more release of glucose from the liver to the circulation. The question is that from where the liver is bringing glucose? Answer is very simple. If it has already stored glycogen, then it will break down the glycogen into glucose. So it means that T3 T4 will lead to increase glycogenolysis, breakdown of the glycogen or glycogenolysis. This is one process going on here. Number two, liver cells will try to make more glucose from non-carbohydrate sources, for example from fatty acids, and that glucose which appears from there, right, for example glucose number one was due to, yes, glycogenolysis, and this second source of glucose is coming from again from the liver, but the source of metabolic pathway which is source of this glucose is gluconeogenesis. It means in the patient's blood, why glucose level goes up? Number one, yes, you know that glucose is coming from GIT more. Okay. Number two, we can say glucose is coming from glycogenolysis. Number three, we can talk about glucose is coming from gluconeogenesis, right? It means that glucose level is really good in circulation. But it's not enough to bring more glucose in circulation. More glucose should be transferred to the cells as well. It means capability of cells to utilize glucose should be enhanced so that cells can produce more ATP. And if cells have to enhance their glucose utilization, they have to take up more glucose. So again, let's recap what we have discussed up to now. Step number one, absorb more glucose. Step number two, yes, increase, what was that? Glycogenolysis. Step number three, increase gluconeogenesis. And step number four, whatever glucose is coming, it should be, yes, step number four, it should be pushed into cells for utilization. And of course, as a good doctor, you should not forget, here there should be help of pancreas, which is bringing, yes, what is it? Insulin. And this insulin help in utilization of glucose. So it's so easy to understand. Look here, let's repeat. What is happening? T3 T4 is more genes are stimulated, more messenger RNAs are made, more proteins are made in the cells. Sodium potassium ATPases are working more, they need more ATP, mitochondrial number increases and mitochondrial size increases. When all this energy requirements increase, we have to provide more fuel to our metabolic machinery. One of the fuel is glucose. To provide more glucose, cell has to take up more glucose. If cell have to take the more glucose, insulin should have. Am I clear? Then not only this, then blood level of glucose should be high so that constant supply of glucose should be there to the cells. For that purpose, GIT absorption of glucose is more and glycogenolysis is more, gluconeogenesis is more. Am I clear? After having understood this carbohydrate metabolism, let's talk about fat metabolism. Now what really happens in the fat metabolism, right? Okay, let's draw a adipocyte here. This is a cell loaded with fat, and let's suppose this is glycerol with 3 fatty acids. This is glycerol with 3 fatty acids. As you know, in the beginning of the lecture, I told you that T3 T4 increase the expression of beta receptors. Beta 1 receptors are more on the heart. Beta 1 receptors are normal, modified beta 1 receptors, which are called beta 3 receptors, which are present more on adipocyte. In the same way, beta 2 receptors are present on, what is this? Hepatocyte. All these receptors are increased. Due to that reason, adrenergic drive is more. And why there is increased glycogenolysis? Because beta 2 receptors are expressed more on hepatocytes, and adrenaline, which is normally present in the body, epinephrine, can drive more glycogenolysis. In the same way, when T3 T4 is more, it acts on adipocyte, right? And increases the expression of beta 3 receptor, and epinephrine can work on that and lead to lipolysis. And when lipolysis will occur, naturally, hormone sensitive lipases will work, and lipolysis will occur. What will happen? There will be release of free fatty acids. So what really happens that in these patients, free fatty acid level in the blood goes down or up? Up. Free fatty acid concentration goes up. And when free fatty acid concentration goes up, this will also add to the, what is this, gluconeogenic pathway, so that more glucose is present, plus more free fatty acid is also present for the tissues which want to do beta oxidation of the free fatty acid for the energy purpose. So what we have seen up to now, that T3T4 has been so clever that it has provided, when it is taking the metabolic rate up, it is providing more fuel like glucose and free fatty acids to the tissues. Is that right? With that, I would like to refer to other plasma lipids as well. Of course, we have already discussed that if T3 T4 is in good concentration, then free fatty acid levels are going up. Is that right? But there are other lipids also, right? In the blood there's LDL, low density lipoproteins, then the phospholipids may be there, and then there may be triglycerides. Now listen. Actually, especially about the hepatocytes, that in the presence of T3 T4, the receptors for LDL increase. Due to that reason, the capability of hepatocytes to take up the LDL is increased. When hepatocytes are taking up from circulation more LDL, LDL is a carrier of cholesterol. It means hepatocytes are clearing the cholesterol more effectively under the direction of T3 and T4. Again, let me repeat. When T3 T4 act on the nucleus of hepatocytes, they work on many genes. One of the gene is, yes, one of the gene is which is making receptors for LDL, and LDL uptake become more. When LDL uptake become more, naturally you must be knowing, when more cholesterol is going within the hepatocytes, de novo synthesis of cholesterol is reduced, and extra cholesterol is going through the bile out of the body. It means plasma cholesterol level will go down. So actually, lipid metabolism is coordinated in such a way that even though free fatty acid level go up, but actually LDL levels in the plasma go down, phospholipid level go down, triglyceride level go down. Now it's too easy to talk about what? That if thyroid hormones can reduce the level of LDL in the blood, it means in patients with hypothyroidism, patients who have chronically low level of circulating T3 T4, their LDL level will be low or high? High. If someone is unfortunately suffering with hypothyroidism, in such patient, LDL cannot, of course, liver will be expressing less receptors for the LDL. LDL concentration or cholesterol concentration in the blood will go up, and you will be very sad to know that patients who suffer with hypothyroidism for a long time, chronically, when cholesterol is high in the blood, they develop accelerated phases of atherosclerosis, and they may die of atherosclerosis related diseases like ischemic heart disease or stroke or other complications. Am I clear? Is there any question about it? Then the other important facts, that not only there is a change in carbohydrate metabolism and lipid metabolism, one thing which is extremely important is, yes, you see, so many enzymes are working less or more. Now you see, carbohydrate handling enzymes are overworking, lipid handling enzymes are overworking, and many enzymes or their coenzymes need vitamins. They need Vitamins, it means it is another duty of the GIT to provide extra vitamins to circulatory system. Is that right? Because there's no fun in talking about increased BMR. It's very easy to understand. If anything which increases basal metabolic rate, it means it increases the function of enzymes and coenzymes. And whatever increases the activity of enzymes and coenzymes, it means that increased basal metabolic rate requires more fuels, it requires more oxygen, and of course, it requires more efficient removal of waste product. And of course, very good doctors will never forget that when metabolism is fast, the enzyme and coenzymes which are accelerating the metabolism, right, they require a lot of vitamins because many vitamins are working as cofactors. Is that right? So that is why in hyperthyroidism, patient may develop relative deficiency of vitamins. Is that right? So anyway, what I really wanted to highlight that in these patients, when T3 T4 is there, vitamin needs is more, right? Then we can come to okay, body weight. What happens if your T3 T4 level goes up? What happens to your body weight? Again, body weight is maintained by a very special thing. What is that? There are so many factors which control your body weight. One of the factors is the balance between catabolism and anabolism. Small amount of T3 T4 along with the growth hormone and insulin act as anabolic hormone. But very high amount of T3 T4 increases the metabolism of the body so much, and there's so much need of fuel in the body that body start breaking its own proteins and own fats to provide the fuel to dump the breakdown product and fuel for the metabolic machinery, and that leads to rapid weight loss. So today onwards, you must remember, if patient has hyperthyroidism, if patient has chronically high level of T3 T4, it means chronically his basal metabolic rate is high, and if basal metabolic rate is high, then patient cells need lot of what? Yes, fuels, building blocks. So from where so much glucose and amino acids and fatty acids will come? Of course, they should come from GIT. But when someone has hyperthyroidism, GIT is hypermotile. And when it is hypermotile, in hyperthyroid patient, GIT is hypermotile, so it does not digest and absorb well. So in spite of the fact that patient has more appetite and hyperthyroid patient is eating more and more, but he is not digesting and absorbing well, and rather he develops diarrhea. Is that right? So what really happens? When he is not getting good amount of nutrients from the GIT, he start catabolizing his own proteins and own lipids. That is why the people with hyperthyroidism undergo catabolism of the proteins and undergo extensive lipolysis, and they may become bony, skinny, and they lose their weight. It's a very important clinical saying that patients who have hyperthyroid, they have a good appetite, rather they may have increased food intake, and in spite of increased food intake, they're progressively losing the weight. Let me explain how it happens. That in patients who are hyperthyroidism, body metabolism is very fast, and oxidative phosphorylation processes may be disrupted in the mitochondrion, and lot of you can say glucose is required, lot of amino acids and fatty acids are required for gluconeogenesis, and lot of fatty acids are required for the beta oxidation processes. But if GIT is unable to provide so much fuel to the body. Why GIT is unable to provide? Because even though person is all the time hungry, and hyperthyroid patient usually eat a lot, but in spite of eating a lot, and in spite of producing a lot of digestive juices from the GIT, but because GIT is hypermotile, so digestion and absorption processes, there's not enough time for proper digestion and absorption of the nutrients from the GIT. So nutrients are wasted, right? Patient develops diarrhea in hyperthyroidism, and nutrients are wasted. So what happens? That metabolic processes lead to excessive breakdown of proteins and lipids, so that provide the, you can say, more and more fuel for the burning metabolic machinery in the body. And that is why you have to remember that usually the patient with hyperthyroid will have high weight or low weight, right? They will lose the weight, and of course it's not a very decent way to lose the weight. Secondly, other way around is also true, that a patient who has low T3 T4 level, if a patient has low T3 T4 level, it means catabolism of protein and lipid is very slow. catabolism of lipid and proteins is very slow, so he will keep on accumulating the things in his body and he will tend to increase weight. So hypothyroid patient tend to increase their weight. Of course, they develop also constipation, right? So hyperthyroid patient develop the diarrhea and lose the weight. Hypothyroid patient develop the constipation as well as they increase the weight. Don't tell anyone that because they develop constipation, so the weight is more. The weight is not due to fecal matter. Weight is more because they're accumulating a lot of proteins and lipids and cholesterol, and even they're accumulating a lot of, you can say, glycosaminoglycans and other substances under the skin throughout the body, right? And those substances may lead to mixed edema, non-pitting edema. Is that clear? Am I clear to you? Again, hyperthyroid patient burning too much calories, right? Even burning its own protein and lipids, weight loss. Hypothyroid patient, very little catabolism, right? And weight gain. Is that clear? Now, we have to talk about some other very important, yes, we should not forget to talk about your heart or patient's heart. What happens to the patient heart under the influence of thyroid hormones? As I told you, thyroid hormone can do many functions. One function of thyroid hormone in the heart is that thyroid hormones increase the expression of beta 1 adrenergic receptor. Number two, thyroid hormones increase the expression of myosin, right? Myosin proteins, and even they increase the expression of genes which lead to increased calcium ATPases. In a nutshell, what really happens that, look, what is happening in the body? Metabolism is low or fast? If metabolism is fast, if T3 T4 is in good amount, metabolism is fast. You need more oxygen or less oxygen? You need more nutrients to circulation or less? So it means arterial flow should be more to the tissues so that more fuel should be coming and more oxygen should be coming. At the same time, venous drainage should also be fast so that from the tissue carbon dioxide and other waste products should be eliminated. So you need faster circulation or slower circulation. If faster circulation you require, then heart has to be slow or fast. So heart has to provide with low cardiac output or fast cardiac output. Fast cardiac output. So what really happens, again let me coordinate for you, that when metabolism goes up, tissues require more oxygen and more nutrients and need to wash away the waste products more efficiently. For this purpose, circulation has to be hyperdynamic, right? You need a faster circulation, right? For faster circulation, heart has to increase its cardiac output. So T3T4 actually increase the cardiac output, heart rate increases, as well as heart rate increases, it means there's positive chronotropic action. In the same way, contractility of myocardium also increases, right, due to changes in the myosin and changes in the calcium handling in the myocytes, and that lead to increased positive inotropic action. When there's positive chronotropic action, that is increased heart rate, and positive inotropic action when there is increased contractility, it means cardiac output will go up. So what really happens with the heart, that there is increased heart rate into increase, yes, stroke volume, that end up into increase cardiac output. Of course, when cardiac output goes up, cardiac output determines the systolic blood pressure. So there is increase in systolic blood pressure. So this is what is happening on the cardiac end, right? Opposite to that, when we talk about what is happening to the arterial side, look, basal metabolic rate is more, there's more heat generation in the body. This more heat generation in the body will dilate the arteries. Secondly, more waste products, more carbon dioxide, more lactic acid and other waste products, they will also dilate the vessels. It means arterioles in the body are dilated. When arterioles are in the body are dilated, it means the peripheral resistance is less and blood will more efficiently move through the circulatory circuit. Is that clear? More rapidly moves through the circulatory circuit. But diastolic blood pressure depends on total peripheral resistance. So what we can say, in these patients, cardiac output increases and systolic blood pressure is high and total peripheral resistance decreases. Total peripheral resistance decreases due to dilatation of most of the arterioles in the body due to increased metabolism, more heat generation, and more waste product generation. Am I clear? And when total peripheral resistance decreases, it means there's decrease in diastolic blood pressure, right? It means the person who was having systolic blood pressure of 120 and diastolic blood pressure of 80, suppose 120 and 80, systolic will go up, it may become 140, and diastolic may go down and it may become 60. What does it mean? That mean blood pressure may not be changed much, but pulse pressure has been changed. The difference between systolic and diastolic has been changed. Previously it was 120 by 80. Now after cardiac output has gone up, systolic has gone to 140, and when peripheral vasodilatation has occurred, total peripheral resistance has come down, diastolic blood pressure has come to 60. So pulse pressure is increased, but mean arterial pressure is not significantly changed. So in a nutshell, if someone asks you what happens to cardiovascular system, what is the role of T3 T4 in cardiovascular system, you will say that T3 T4, because it increases the metabolism in the body, so it has to make the cardiovascular system more efficient, so that tissue should be provided with extra cardiac output and perfusion, right? For that purpose, heart rate increases, stroke volume increases, systolic blood pressure increases, as well as most of the peripheral vessels dilate, so that more blood should go to the tissues and diastolic blood pressure falls. Am I clear to everyone? Right? Now we come to another thing. Of course, you should not forget about the oxygen supplier, an eliminator of carbon dioxide, your lungs. Of course, if a patient has high metabolism, oxygen demand is more and production of carbon dioxide is also more. It means that lung has to provide more oxygen to the blood and eliminate more carbon dioxide. Lungs has to provide more oxygen and lung has to eliminate more carbon dioxide. So what does it mean? Lungs have to be more efficient. For this purpose, again listen carefully. What will happen? There will be increased respiratory rate as well as increase, yes, tidal volume. And when respiratory rate will increase and tidal volume will be increased, it means minute ventilation is increased, right? And when minute ventilation increase, it means there will be increased what? There's increased supply of oxygen to the tissues, at the same time better elimination of carbon dioxide from the body. So it means respiratory system is also doing its best. Now again, let's recap up to this, right? What we have discussed. Listen carefully. Let's go logically. T3 T4 hormones are not just funny hormone doing unrelated things. T3 T4 hormones are part of the master plan in the body to maintain the metabolism as well as to maintain the maturity and growth. Now listen. From the very beginning, what happens? T3 T4 is excess. Metabolism, what will happen? From the very beginning, many genes stimulated, lot of new proteins made, lot of sodium potassium ATPases made, lot of enzymes made, mitochondria become more in number and size, metabolism in the tissues goes up, tissue require more nutrients and they release more waste product. Is that right? So the body, multiple areas have to function, multiple systems in the body has to function in such a way to provide more fuels and eliminate the waste. For example, to provide more glucose, appetite will increase, GIT will absorb more glucose. Is that right? In the same way, glycogenolysis will occur, gluconeogenesis will occur, and this extra glucose which is coming to push into cell, insulin is extra produced. Am I clear? Meanwhile, lipolysis start. Again, more fatty acids are provided so that fatty acids can be used directly for the energy purpose or their skeleton can be used for gluconeogenesis. Am I clear? Then, special handling of the other lipids, that when T3 T4 is more, liver cells express more LDL receptors. So cholesterol is more efficiently cleared from the blood and blood level of cholesterol goes down. Even other associated lipids like phospholipids and triglyceride level also in the blood go down. Am I clear? With that, we need more. Yes, what we need more? We need more cardiac output so that more circulatory system should become more fast and supply of the nutrients and oxygen is more efficient and removal of the waste product is as well more efficient. To increase the cardiac output and circulatory speed, what will happen? Heart rate will go up, stroke volume will go up, of course cardiac output will go up, and eventually that will translate into increased systolic blood pressure, right? During the same time, when body heat is generated more, body heat is generated more, so arterioles dilate and total peripheral resistance decreases, and when total peripheral resistance decreases, diastolic blood pressure falls. As systolic goes up and diastolic falls, mean arterial pressure is not significantly changed, but pulse pressure is really changed very significantly. Then of course you should not forget, not only we need more cardiac output, we need also more oxygen. For that purpose, respiratory system itself is stimulated, and there is tachypnea with more depth of respiration. So rate of respiration is more, depth of respiration is more. It means that rate of respiration and tidal volume both are more. Eventually minute ventilation is more. Advantage of this change in the respiratory system is more oxygen can be supplied to the body more efficiently, but more importantly, there is better elimination of carbon dioxide, which is produced due to high metabolism. Once we have discussed, of course, we should not underestimate the GIT, which is a major fuel provider. Of course, if there's T3 T4 level very high, GIT will undergo diarrhea, and what will happen? Due to hypermotility, it will go into diarrhea, then absorption of the, what is that? Fuel will be less, right? So excess of T3 T4 is not good, right? Another thing which is important, there's so many enzymes are working in the body, metabolism is so fast. These enzymes or coenzymes require a lot of vitamins, so relative deficiency of vitamin can also develop. Now we come to some special actions, right? For example, effect of all this T4 activity on your great central nervous system. You have a great central nervous system, am I right? Sure. That is why now you will tell me, what is the action of T3T4 directly on central nervous system? Does it increase PMR or not? Answer is no, right? The T3T4 does not directly increase the basal metabolic rate in central nervous system, but still it produces many actions in central nervous system. Why? Because T3T4 expresses more adrenergic receptors in central nervous system, and many other receptors and neurotransmitter activity is altered. So when T3T4 is more, CNS is also more functional. That is why people who have hyperthyroidism, their central nervous system actions become more, they look more alert, even they look very much anxious, and they cannot sleep well in spite of the fact that they are very tired due to hypermetabolism. They are very weak and tired, but due to increased synaptic activity in central nervous system, these people develop insomnia, difficulty to have enough proper sleep. Is that right? So if someone who has hyperthyroidism, he will develop insomnia, he will develop difficulty in breathing, sleeping, that is insomnia, he will develop anxiety, he may develop even paranoia, he may start doubting on everyone. Is that right? Inappropriately, right? So what I'm talking about, that there will be bad effect on the central nervous system if there is excessive amount of T3T4. Of course, other way around is also true. If T3T4 level is less, central nervous system will become what? Slow, right? And then so if you develop deficiency of T3T4 in childhood, mental maturity is not achieved. But once you have achieved mental maturity, and after that hypothyroidism develop, then what really happens is central processes become slow. So what happened? There's lack of concentration in hypothyroid patient. There's excessive sleep in hypothyroid patient, and hypothyroid patient due to slow mental mechanisms even may become depressed. So if there's anxiety, it is hyper. If it's depression, it is hypo. If patient is more talkative and someone tells you he has severe thyroid disturbance, it must be hyper. If patient is not willing to talk at all, or depressed, or very slowly talking in a low voice, it must be hypo. Is that right? If patient is sleeping less, it is hyper. If patient is sleeping more, it is hypo. If patient has diarrhea, it is hyper. If patient has constipation, it is hypo. If patient is having more cardiac output and tachycardias and pulse rate is very high, even sleeping pulse rate is high, it is hyperthyroidism. And if it is slow, it is hypo. But remember one interesting thing. If there's severe hyperthyroidism, heart may fail when it cannot provide the extra cardiac output demands. Or even severe hypothyroidism, heart may fail due to very poor contractility and very poor heart rate, right? Now, another thing, of course, if weight is lost, we are talking about hyper. If weight is being gained, we are talking about hypo. Is that right? Now, another thing, effect of T3T4 on the hormones, other hormones. It's very easy to understand that if T3T4 is more, then we need other hormones in excessive amount. For example, simply talk about insulin. If T3T4 is more, you need less insulin or more? You need more insulin to handle more fuel in the body, especially carbohydrate. In the same way, if T3T4 is more and you are building the bones, then you need more parathyroid. But remember, if T3T4 is excessively more, then it may lead to catabolism. Right? Now, last but not the least, now we are going to talk about some very important organ system in the body. What is that? You are a young man, you must be interested in that system. Yeah, reproductive system, isn't it? Reproductive system. What are the effects of TCT4 on the reproductive system? As I told you previously, that of course you can recognize this structure. Am I right? Right? There's no fun in going into detail. Right? Uterus is there and vagina is there. Fallopian tube with ovaries. Now, what really happens? Remember one thing, that T3, T4 do not increase the metabolism, basal metabolic rate of gonads. They do not increase the basal metabolic rate of your ovaries, I mean female's ovaries, or increase the metabolic rate of uterus, or in the male, testes metabolic rate is not increased. But because there is disruption with when T3T4 levels fluctuate up or down, there's hyperthyroidism or hypothyroidism that disturb hypothalamus as well as interior pituitary. So hypothalamus, pituitary, gonadal axes are disturbed, right? For example, in female, hypothalamus, pituitary, and ovarian cycle is disturbed, axis is disturbed, so hormonal functions are not well. Is that right? So what happens to a female? Number one, if there's hyperthyroidism or hypothyroidism, in both cases she loses the libido. Number one. Is that right? Number two, her menstrual cycles are disturbed. Usually, in hyperthyroidism, she develops oligomenoric. Oligomenorrhea means less frequent menstrual bleeding, right? And in hypothyroidism, usually there's menorrhagia, that is excessive menstrual bleeding, or there's polymenorrhea, more frequent menstrual bleeding. And with that, there may be infertility. That is why one of the very important investigation in case of looking for the cause of infertility is do the T3, T4 level and TSH level, because hyperthyroidism or hypothyroidism, both hyper or hypo, both can make the person infertile by disrupting the, especially in female, by disrupting the hypothalamic, pituitary, ovarian axis. Is that right? Now, if we talk about the males, their nature has not spared them even. Again, don't tell anyone that T3T4 excess will increase the metabolism or BMR of the testes or low will decrease. BMR of the testes is not dependent on T3T4, but still testes are dependent on hypothalamic pituitary axis. Is that right? Right? So what really happens? They are also disrupted. So males also lose the libido and they may also become infertile or impotent, especially in hypothyroidism when they are very depressed. Depressed male are usually impotent. Cause of depression may be anything, including hypothyroidism. Is it clear to everyone? So that's all about the basic concepts related with the function of functions of thyroid hormones. Again, let's recap. Thyroid hormone, number one, act on many genes and increase their expression. Secondly, thyroid hormones increase metabolic activity by increasing sodium potassium ATPase activity, increasing number and size of mitochondria. Thyroid hormones are very important for physical and mental maturation, right? Then thyroid hormone act on many specific bodily mechanisms, all of these to provide more nutrients in the body and to eliminate the waste products due to high metabolism. Now, my dear students, we are going to talk about thyroid hormones and gland and its clinical correlates, right? We'll especially concentrate on the concepts of hyperthyroidism and hypothyroidism. But remember one thing, that this pathophysiology or especially hyperthyroidism and hypothyroidism will be discussed in detail in pathology lecture. Here we will just briefly review some relevant ideas which are relevant with physiology. For example, in hyperthyroidism, what is hyperthyroidism? Hyperthyroidism is a clinical pathological condition in which patient has chronically elevated T3 T4 level in his body or tissue, right? And what could be the causes of it? There's so many causes, but common causes are Graves' disease. It's an autoimmune disease which is more common in females. Toxic thyroid adenoma, multinodular toxic goiter, exogenous administration of T4, right? These are few important causes. Let's concentrate on what's Graves' disease. What really happens in Graves' disease, that this is an autoimmune disease which is more common in females, and immune headquarters produce autoantibodies which are directed against the TSH receptors of the follicular cells. So these autoantibodies which are coming from the immune system, these autoantibodies attack the TSH receptors. When these autoantibodies attack the TSH receptors, not only these autoantibodies bind with the receptor, but they also stimulate the receptor. Let's focus here. What we have to learn, that not only antibodies bind with the receptor, but they also stimulate the receptors. And when they stimulate the receptors, it means these autoantibodies behave like TSH, right? But the difference in TSH and these autoantibodies is that these autoantibodies are able to stimulate the receptors for more powerfully and for longer duration as compared to TSH, right? Now, normally what happens, all of you know that from the hypothalamus there is a production of TRH, and this TRH goes to interior pituitary and lead to release of TSH, right? Hypothalamus is producing TRH, and this TRH is going to the interior pituitary through portal system and stimulate that release of TSH. And normally this is a TSH which is supposed to stimulate this receptor, right? Now, as I told you, in these patients, because these autoantibodies stimulate the gland very powerfully, these autoantibodies are called thyroid stimulating immunoglobulins. And because not only they stimulate the thyroid cells, but they can lead to the growth of the thyroid cells, so they are also called thyroid growth immunoglobulins. So these thyroid stimulating immunoglobulins and thyroid growth immunoglobulins, which bind with TSH receptors, not only they increase the function of the cell, but they also lead to the growth of the cell and proliferation of the cell, right? So what are the results of this? Result of this is that there is excessive production of T3 and T4, right? And this T3 T4 which is going to the circulation, right, as you know, this T3 and T4 will eventually produce its metabolic actions on the peripheral cells, right? And what will really happen, that BMR will be, basal metabolic rate will be increased in all tissues. Of course, you know that BMR is not increased in gonads and the brain, spleen and the lymph nodes, but all other tissues BMR is increased. Secondly, body has, you can say, more responsiveness to adrenergic drive. You know, beta adrenergic receptors are also expressed in excessive number in the tissues under the directions of T3 and T4. With that, right, now again let me tell you what's really happening, that immune system is producing autoantibodies. These autoantibodies are stimulating TSH receptors, and they are not only binding, they are stimulating for stronger than the normal TSH, and forcing these cells to produce more T3 T4. Not only forcing these cells to produce more T3 and T4, along with that, these also lead to the growth of the cells, and these cells with the time become columnar and they become more in number, right? So follicles become crowded with the cells, and you can say, due to excessive, in Graves' disease, due to excessive stimulation of TSH receptors, cells undergo, cuboidal cells become columnar cells, and there is excessive number of cells per follicle, and even total number of follicles also increase, and eventually there is diffuse enlargement of thyroid gland. Thyroid gland enlarges in every portion, and this diffuse enlargement of thyroid gland is called diffuse goiter. Again, let me make a very clear concept. The goiter means there is a large thyroid, but goiter does not tell you, if I say this patient has goiter, it does not tell you he is hyperthyroid or hypothyroid or euthyroid. Remember one thing, goiter simply means that person has a thyroid which is large, but a goiterous thyroid gland, which is large thyroid gland, may be hyperfunctioning or maybe normally functioning or maybe hypofunctioning. So let me tell you here, if we talk about goiters, some goiters are hyperfunctioning. Is that right? Goiters are associated with hyperthyroidism, and some goiters are having normal T3 T4 production. We call them goiters associated with euthyroidism, and some goiters are associated with low production of T3 T4, and we call them they are producing hypothyroid situation. What I really want to put in your mind is goiter tells you about the size of the gland. It does not tell you about the function of the gland. It's quite possible one patient has goiter with hyperthyroidism, other patient has goiter with euthyroid state, and still another patient who has goiter with hypothyroid state, right? But when we talk about the Graves disease, right, this is also a goiter but without any nodules. There is diffuse enlargement of goiter, right? Diffuse enlargement of the gland. Because it is diffuse enlargement of the gland, it is a goiter, but it is diffuse goiter, and with that there is thyrotoxic state, because this goiter is producing excessive amount of T3 and T4. Is that clear? Now, what will happen to this person? This person will develop thyrotoxic state. Person will develop thyrotoxic state, thyrotoxicosis. Thyrotoxicosis means the clinical features related with the hyperthyroidism and complications of hyperthyroidism. I will go into detail of thyrotoxicosis later. So number one, these patients, again let me repeat it, immune system overstimulates, autoantibodies overstimulate thyroid tissue, that leads to cells which are cuboidal become columnar, plus there is increased number of cells per follicle, and total number of follicles is also increased, so total size of the gland is increased, right? Diffuse goiter is there. Along with that, massive amount of T3 T4 is produced, and this T3 T4 produces the clinical features of thyrotoxicosis. Along with this T3 T4 produces a negative feedback mechanism, and that will suppress the production of TSH as well as production of TRH. So in these patients who are having Graves disease, they are having low TRH and of course low TSH level, circulating TSH levels, right? Now, there are two more important points, that this immune headquarter, these antibodies may also attack the extraorbital connective tissue, right? This is your eyeball, right? There are extraocular muscles here, and there may be some fat and connective tissue. In some unfortunate patients, these autoimmune processes not only attack the thyroid tissue, but these autoimmune processes also attack the extraocular, right, retroorbital connective tissue. So extraocular muscles may be attacked by these antibodies, right? Some people believe that extraocular muscles and connective tissue or retroorbital fat, these may have some antigens which are structurally similar with the TSH receptor. Due to that reason, those autoantibodies attack here. And those autoantibodies attack over here, this tissue is also get injured and inflamed. And when the tissue becomes injured and inflamed and edematous and swollen, so it means that connective tissue amount in the orbit will increase, and this will push the eyeball forward. So eyeballs will be protruding forward and outward, and this condition is called exophthalmos. This condition is called exophthalmos. So what we can say, that these patients who are having thyrotoxicosis, right? Number one, immune system attack classically on the thyroid tissue, and produces diffuse goiter. Along with that thyrotoxic state, by increased secretion of T3 T4, of course that suppresses TRH and TSH levels in the blood. Number two, this immune system may attack the connective tissue in the orbit, and that may lead to inflammation of the connective tissue and excessive production of proteoglycans and edematous status. And this inflammation and edema in the orbital connective tissue lead to it squeezes the eyeballs forward, and that produces exophthalmos. And in some unfortunate patient, the same, you can say, autoimmune processes may attack some skin over the lateral malleolus, right? And over the lateral malleolus, right, if the skin is having autoimmune attack, that may lead to some thickening of the skin over here and some undue deposition of connective tissue with some water absorption over this, and we call it pretibial myxedema. Please don't confuse pretibial myxedema with the generalized myxedema which is present in hypothyroidism. Again, let me repeat it. This is a confusing term, myxedema. Generalized myxedema occur in a patient who are suffering with hypothyroidism, right? Where all over the body, under the skin, there's a heavy deposition of connective tissue and water absorption of that, generalized myxedema. But in patient of Graves' disease, sometimes we see that there is pretibial myxedema, especially slightly above the lateral malleolus, right? So this was something about the Graves' disease. I have not discussed the features of thyrotoxicosis. That I will discuss with the after finishing these causes. Now, already you know about the Graves' disease. That Graves' disease is an autoimmune disease. Let's recap. It's an autoimmune disease. It's more common in the females. Male to female ratio is 9 is female to male ratio is 9 is to 1. It is more common in the age of 30 to 50. Graves' disease autoantibodies are classically directed against the TSH receptors on the follicular cells. Follicular cell increase the synthetic and secretory activities, right? Due to that reason, thyrotoxic state occur. Number two, follicular cell may undergo growth pattern and eventually they undergo proliferation. That may lead to formation of diffuse goiter. Because this goiter is associated with hyperthyroidism, we can also call it diffuse toxic goiter in Graves' disease, right? Of course, you must know that this T3 and T4 which is produced, that will act on the thalamus and hypothalamus, reduce the TRH production, and it powerfully act on the anterior pituitary, and by negative feedback inhibition, this T4 will inhibit the production of TSH. So in Graves' disease patient, what really happens that T3 level is up, T4 level is up, TSH level is low, TRH level is low, and in the blood, thyroid stimulating immunoglobulins and thyroid growth immunoglobulins can be detected, right? Now let's talk about the toxic thyroid adenoma. Art is you must be able to differentiate them. As a good doctor, you must be able to differentiate both of them. Graves' disease was an autoimmune process, but this thyroid adenoma is a neoplastic process. It's a new growth. It's a benign neoplasia, neoplastic process. What really happens into this? Actually, what really happens here is that let's suppose this is a thyroid gland, right? And it is full of lot of follicles which are normally functioning. Sometimes it happens in some patients, unfortunately, one particular cell undergo neoplastic transformation. Then that follicular cell lead to formation of adenoma. And this adenoma is able to take the radioactive iodine or simple iodine, and this adenoma is functional. Because this adenoma is functional, this adenoma cells produce this is autonomous adenoma, right? It means its activity is not dependent on TSH, right? And this adenoma produces massive amount of thyroid hormones. This adenoma produces massive amount of T3 and T4, right? And that produces severe, you can say, toxicity, thyrotoxicity. Again, process is the same. When T3, T4 is so much produced, what really happens? That TRH level goes down in the same way TSH levels go down. When TSH levels go down in these patients with toxic adenoma, TSH levels go down, right? Because they are inhibited by circulating high levels of T3 T4. When TSH levels go down, then TSH is a normal drive for most of the thyroid tissue, right? So what really happens? This adenoma keep on producing massive amount of T3 T4, and this massive amount of T3 T4 produces thyrotoxicosis or clinical features of hypothyroidism. Along with that, this T3 T4 suppresses the production of TSH, and due to absence of the TSH, right, the normal thyroid tissue is deprived of its normal stimulatory action. So what really happens? The normal thyroid tissue undergoes hypertrophy. So what really happens is you find there is a very small gland attached to this adenoma, right? A trophic gland. Oh, please don't confuse this with the Graves. Look, what was happening in Grave? Grave was an autoimmune disease. Toxic adenoma is a neoplastic disease. It's a new growth, benign usually. Of course, adenomas are benign. Number one. Number two, Graves disease is more very common in females as compared to the males. This is not true about toxic adenomas. Number three, in Graves disease, there's diffuse goiter. There's generalized diffuse enlargement of thyroid gland. Here there is one nodule which is very large and hyperfunctioning, and rest of the gland is shrunken, right? But there are few things common. In both cases, T3 T4 levels are high. Both cases have clinical features of hyperthyroidism. Both cases have low TSH, but Graves disease has autoantibodies directed against TSH receptors, but adenoma does not have autoantibodies directed against TSH receptors. Am I clear? Then we can come to one more cause, multinodular toxic goiter. This type of problem occurs in patients where there is iodine deficiency, right? Due to iodine deficiency, what really happens that people who are living in such areas where there is iodine deficiency, that may that normally lead to hypothyroidism. But in some people, when there's iodine deficiency, what really happens that some follicular cell keep on working well, others are not working well, and TSH level initially goes up, and when TSH level goes up, it overstimulates some of the follicular cells, and that end up into autonomous growth formation or hyperfunctioning thyroid areas. So we can say that multinodular goiter may become toxic, but remember, iodine deficiency usually or more commonly lead to hypothyroidism. Then in the same time, we can discuss that people who are taking thyroxine tablets, and if they are taking thyroxine replacement due to previously diagnosed hypothyroidism, if they are taking excessive doses, then naturally they may develop the clinical features of thyrotoxicosis, right? Now these are few causes related with hyperthyroidism, but we did not discuss the clinical features of hyperthyroidism. Now let me tell you what really happens in a patient with hyperthyroidism. Okay, let's suppose I have the hyperthyroidism. If I have a hyperthyroidism, I have a thyrotoxic state. It means my circulating level of T3 T4 and tissue activity, you can say T3 T4 activity on the tissues is very high. I may have due to Graves disease. I may have this problem due to toxic nodular goiter, or I may have this problem due to some, you can say, toxic multinodular goiter or toxic adenoma, or I may have this problem unfortunately if due to any reason I'm taking thyroxine inappropriately in high concentration. So all these conditions may lead to thyrotoxic state, right? Okay, let's take a classical case. A patient come to you and patient is having Graves disease. Now what will happen? Number one, from head to toe we will work up. When T3 T4 levels are very high, proteins catabolism start because basal metabolic rate is very high. Body need more fuel, right? It has to burn more calories, right, to maintain high T3 T4 dependent basal metabolic rate. So of course all the proteins are undergoing catabolism, and when this hypercatabolic state, your hair become thin. So don't look at my hair, they are lost. But actually, if a patient is hyperthyroid, the patient's hair become thin, right? Number one, in hyperthyroidism. But let's compare it with the hypothyroidism also. Okay, right? Let's compare it with the hypothyroidism also. In hyperthyroidism, hair are lost or hair become thin. In hypothyroidism, hair become thick and unmanageable, right? Then another thing. which is very important in hyperthyroidism, because activity in central nervous system is more, right? In central nervous system, adrenergic receptors are more, and central nervous system activity is more. So patient looks anxious. Patient looks have a worried look, right? But when you compare hyperthyroid patient with hypothyroid patient central nervous system is slow, so patient looks depressed. So again, anxiety is seen in a patient with hyperthyroidism, and depression is seen in a patient with hypothyroidism. Is that right? Then we come to the next thing, that insomnia. Person whose central activity is very high, central nervous system activity, person cannot easily sleep. So this is very a big problem to have a proper sleep. So these patients who are hyperthyroid, they have insomnia. Opposite to that, people who have hypothyroidism, they don't have insomnia, rather they oversleep, right? We call that condition somnolence, right? Again, let's compare. There here the hyperthyroid patient, and here the hypothyroid patient. In hyperthyroid patient, here become thin. In hypothyroid patient, hair become less and thick and unmanageable. In hyperthyroidism, patient looks anxious. In hypothyroidism, patient looks depressed. In hyperthyroidism, patient has insomnia, difficulty to have sleep. In hypothyroidism, patient sleeps all the time, as much you allow him to sleep, right? Then in hyperthyroidism, patient talks more. I don't mean that he gives lecture like me more, but in hyperthyroidism, person is more talkative, right? In hypothyroidism, person is having slow, you can say psychomotor activity. Even in hyperthyroidism, tendon reflexes are more. In hypothyroidism, tendon reflexes are slow, right? In especially tendon reflexes relax slowly. Then in hyperthyroidism, there are tremors, and these tremors are fine tremors in hyperthyroidism, right? Not coarse tremors like cerebral disease or coarse tremor which are seen in Parkinsonism. In hyperthyroidism or thyrotoxic state, they are fine tremors. Then you come down. Let's look at the eyes. Now, patient with hyperthyroidism, will keep on comparing hyper with hypo. Patient with hyperthyroidism, where thyroid hormones are more, right? If hyperthyroidism is especially due to Graves' disease, then eyes may be protruding unduly forward and there may be exophthalmos. Another important thing which I would like to tell you about the hyperthyroidism is, in hyperthyroidism, adrenergic receptors are more in levator palpebrae superioris, and levator palpebrae superioris muscle is working more and it is having undue pull to the upper lid. So when patient eyeballs turn downward, upper lid fail to follow, right? We call this situation lid lag. Again, for example, patient is, if you put your finger in front of the patient like this and you take your finger gradually down, normally when patient eyeball will turn down, right? If patient keeps focused on your finger, as eyeballs are turning down, upper lid also follows the eyeball. But in these patients, because levator palpebrae superioris is overactive, right? So eyeballs do turn down, but upper eyelid fail to follow that. So we call this condition lid lag. Now let's come down. Let's talk about the skin. Skin in case of hyperthyroidism is warm. In hyperthyroidism, skin is warm because basal metabolic rate is more, heat generation is more, so skin has to be warm to get rid of the heat which is produced in the body. But in case of hypothyroidism, skin is usually cold. Then another important thing is that patients who are hyperthyroid, because the body heat generation is more, so they are intolerant to heat, right? They get very upset if they are, especially in the summers or in warm places. But when we talk about the hypothyroidism, these patients are intolerant to cold because their basal metabolic rate in hypothyroidism is slow, so they are unable to, you can say, tolerate the cold environment, right? So again, here the skin in hyperthyroidism, skin is warm and sweaty, and in case of hypothyroidism, skin is thick, skin is scaly, and skin is, yes, and skin is dry, and skin is thicker. Why? Because in hypothyroidism, metabolism in the body is low and catabolism of the proteins is low. So under the skin, proteoglycans and many other like hyaluronic acid and glucuronic hyaluronic acid, proteoglycans and glycosaminoglycans. These substances accumulate under the skin in hypothyroidism, and generalized skin become thick, and with these substances water adsorb, and this condition is called generalized mixed edema, which is the feature of hypothyroidism. And hypothyroidism, due to accumulation of these substances under the eyes, you may find puffiness, right? So the skin changes. And another important thing in hypothyroidism, hypo, because generally metabolism are slow, vitamin carotenes cannot be converted into vitamin A, and carotenes, which are yellow pigment, they accumulate in the body and they produce yellowness in the skin. But remember, how do you differentiate hypercarotenemia with the jaundice? Hypercarotenemia is the feature of hypothyroidism. Again, let me repeat it. In a patient with hypothyroidism, carotenes cannot be converted into vitamin A properly. So carotenes levels go high in the blood and eventually into tissue, and skin gives a yellow tinge. Skin gives a yellow tinge. But very important point is you must differentiate this yellow coloration of the skin due to carotenes from the jaundice. Answer is very simple. Just look in the eyes of the patient, right? Look on the sclera, more truly speaking. Sclera, when there's jaundice, sclera will also turn yellow, but carotene does not turn the sclera yellow. Anyway, let's come back. Okay, very important point. Even the way patient talk, you can tell on telephone. If a patient calls you and I say this patient has hyperthyroidism or hypothyroidism, any one of these conditions, on telephone you can tell. Of course, hyperthyroid patient will be having anxious voice, shrill, but hypothyroid patient has hoarseness of voice. Why the hoarseness of voice is there? Because in hypothyroid patient, even on the vocal cords, a lot of proteoglycans and other substances accumulate and make the vocal cord thick. And due to that reason, in hypothyroid patient, when vocal cord become thick, the voice also become thick, and the face become toad-like, and you can say that they have hoarseness of voice and they cannot talk well, right? So this is about hypothyroidism. Then we come down please from the face. Okay, there's the respiratory system. You know, hyperthyroidism, metabolism is high. In hypothyroidism, metabolism is low. So hyperthyroid patient need more oxygen and release more carbon dioxide. Hypothyroid need less oxygen and release less carbon dioxide. It means respiratory function should be more in hyperthyroid and less in hypothyroid. Due to that reason, you find usually patient with hyperthyroidism tachypneic. But in case of their respiratory rate and depth of respiration may be more than normal. But when we go to the hypothyroid, their respiratory rate may be less than normal, right? Then, of course, let's go to GIT. Yes, here is GIT beginning. All of you know oral cavity. Now we have to see, of course, you know where it extends in the end. I will not go into detail, but right now we'll concentrate what are the effects of hyperthyroidism and hypothyroidism on the GIT. Very simple. Hyperthyroidism will give increased activity in the GIT. So GIT motility will increase in hyperthyroidism. When GIT motility will increase, at the top GIT secretions will increase. But due to increased mortality, transit time to the GIT is less and food, right? So digestion and absorption is not well and patient develop diarrhea. Usually patients who are hyperthyroid, these patients have increased appetite, they eat more, but in spite of that, they keep on losing the weight. Hyperthyroid patients keep on losing the weight because it's simply burning their lipids and burning their proteins, right? That is a hyperthyroidism, a hypercatabolic state. So these people feel more hungry, they eat more, but because GIT is hypermotile, so they are unable to digest and absorb well, so they develop the diarrhea, and in spite of eating a lot, they keep on losing the weight. Opposite to that, when we talk about hypothyroid patient, yes, the GIT is slow. As I told you, hyperthyroid patient have diarrhea, you can anticipate that hypothyroid patient will have, yes, constipation. Is that right? Hypothyroid patient will have constipation, right? Another thing which is very important is hypothyroid patients usually eat less, but still they are having weight gain. The reason being that most of the protein turnover and carbohydrate turnover is slow in the patient with hypothyroidism. Is that right? So we can say This patient, again recap all what we have discussed. Hyperthyroid patient is anxious. Hypothyroid patient is depressed. Hyperthyroid patient is, yes, insomniac. Hypothyroid patient is sleeping a lot. Hyperthyroidism patient, anxious voice. Hypothyroid patient, hoarseness of voice. Hyperthyroid patient, warm flushed skin. Hypothyroid patient, dry scaly cold skin. Hyperthyroid patient, intolerance to heat. Hypothyroid patient, intolerance to cold. Hyperthyroid patient, if it is due to exophthalmos, of course, if it is due to, sorry, Graves disease, patient will have exophthalmos and lid lag, right? Opposite to that, in case of hypothyroid patient, there may be puffiness under the skin. Then, as I told you, that overall weight loss is there in hyperthyroidism and weight gain is there in hypothyroidism. Diarrhea is there in hyperthyroidism and constipation is there in hypothyroidism, right? Hyperventilation is more, respiratory system is overactive in hyperthyroidism, respiratory system is underactive in hypothyroidism. Of course, then we come to the cardiovascular system. In hyperthyroidism, on the heart, beta 1 receptors are more. On SN node, beta 1 adrenergic receptors are more. On AV node, beta 1 receptors are more. So, what really happens? Or so, adrenergic drive on the heart is more. So, heart rate goes up in hyperthyroidism. Hyperthyroid patients are tachycardic patient, and the pulse rate is fast. Even sleeping pulse rate is fast, right? In hyperthyroidism, and cardiac output is more because there is positive chronotropic action as well as there is positive inotropic actions, right? So hyperthyroid patients usually have more than normal cardiac output. One reason is heart is directly stimulated by adrenergic system. Number two reason is that body needs more cardiac output for its higher BMR, right? Especially because tissues are in hyperthyroid patient, tissues are producing more carbon dioxide, producing more lactic acid, and utilizing more oxygen. So tissue lead to arteriolodilatation, because in hyperthyroid patient most of the arterioles in the body are dilated, total peripheral resistance is less, so heart is beating against less resistance, so its cardiac output is further increased. So we can say a patient with hyperthyroidism has hyperdynamic circulation. Patient with hyperthyroidism has hyperdynamic circulation. Opposite to that, if you look at a patient with hypothyroidism, his heart rate is slow, contractility is slow, cardiac output is less, right? So usually patient with hyperthyroidism develop tachyarrhythmias and patient with hypothyroidism develop bradyarrhythmias, right? Another important thing, which is very important, patient with hyperthyroid may also undergo cardiac failure. And here thing is common. Even patient with hypothyroid may also undergo cardiac failure, because hyperthyroidism eventually produces cardiomyopathy. In the same way, hypothyroidism also produces cardiomyopathy. Secondly, hyperthyroidism may precipitate tachyarrhythmias, and hypothyroidism may lead to bradyarrhythmias and sinus bradycardias and AV node blocks. So all these things, in hyperthyroidism there may be high output cardiac failure, and in case of hypothyroidism there may be low output cardiac failure. After that, we can come to, yes, something very, very important. That is, in hyperthyroidism, cholesterol LDL receptors in the liver are expressed more, so cholesterol is more efficiently cleared from circulation. But in hypothyroid patient, hepatocyte express less LDL receptors, so from the blood, cholesterol clearance is poor. Because cholesterol clearance is poor in hypothyroid patient, what really happens? That cholesterol levels start going up. In patients who are hypothyroid, their cholesterol level or LDL level in the blood goes up, and it has very important implication. These patients have, of course, hypercholesterolemia, which lead to accelerated atherosclerosis, and many of these patients may have increased risk of ischemic heart disease or stroke or other atherosclerosis related problems, right? So we have discussed about the hair. Here the thin in hyper, here thick. We have talked about the central nervous system, talked about eyes, we have talked about skin, we have discussed about the GIT, we have discussed about the respiratory system, we have discussed about the cardiovascular system. One important thing in hyperthyroidism, because cardiac output goes up in hyperthyroidism, so systolic blood pressure goes up. But in hyperthyroidism, because arterioles dilate, So total peripheral resistance is less, and you know that diastolic blood pressure depend on total peripheral resistance. So when total peripheral resistance is less, what really happens? Diastolic blood pressure become less. So in hyperthyroid patients, because cardiac output is more, systolic blood pressure goes up, and because total peripheral resistance is less, diastolic blood pressure become less. So systolic goes up, diastolic become less, so pulse pressure increases. What I really mean that if a patient had blood pressure of 120 by 80 millimeter of mercury, after hyperthyroidism, patient may develop a blood pressure of 140 by 60 millimeter of mercury. Right, mean arterial pressure may not change, but systolic and diastolic pressures do change, right, and pulse pressure may increase. Now, after that, we come to another important aspect, that is skeletal muscles, right? Actually, in hyperthyroidism as well as in hypothyroidism, skeletal muscles become weak and patient develops fatigability, right? Then another thing is related with the urogenital system. Yes, right? Males and females both suffer in hyper and hypo both. For example, females who have hyperthyroidism, right? Their hypothalamic pituitary ovarian axis is disturbed, right? And that usually translates into oligomenorrhea. Their menstrual bleeding is less. But in case of hypothyroid female, again, hypothalamic pituitary ovarian axis is disturbed. But this may translate into polymenorrhea or even menorrhagia, right? But one thing which is extremely important, that hyperthyroid state or hypothyroid state both can lead to infertility, right, in the males as well as in the females, right? Then we can come to some other clinical features related with hyperthyroidism and hypothyroidism. I think this is enough. Okay, we were talking about hyperthyroidism. Most of the patients who have hyperthyroidism, they are suffering with primary hyperthyroidism. Primary hyperthyroidism means that cause of hyperthyroidism lies in thyroid gland. Primary hyperthyroidism is, for example, when there is Graves' disease, or there is toxic adenoma, or there is any other condition, toxic multinodular goiter, in which there is inappropriately produced excessive T3 T4. Now, what really happens when, for example, in Graves' disease, when T3 T4 is inappropriately produced excessively, right? This T3 T4 will suppress the production of TSH as well as suppress the production of TRH. So, what will be the result? That T3 T4 levels are high, but TSH level and TRH levels are low. So, what is there in primary hyperthyroidism? That TRH level is high, and this high level of in primary hyperthyroidism, what really happens, T4 level or thyroxine level in the blood are high, and these thyroxine levels are suppressing the, yes, TSH as well as suppressing the TRH. So again, let me repeat, if a patient comes with thyroid toxic state, if a patient comes with clinical features of hyperthyroidism, right, and if his T4 is high, TSH is low and TRH is low, the cause of hyperthyroidism is present in the, most probably in the thyroid gland. This is called primary hyperthyroidism. Now we go to another condition. Let's suppose this is another patient, and this is a less common condition in which there is a problem in the interior pituitary. That in interior pituitary, let's suppose this adenoma or tumor, and this tumor is producing massive amount of TSH inappropriately. This is, such patients are less common, but they are there. That someone has adenoma in interior pituitary or tumor in interior pituitary gland, and this is producing massive amounts of TSH, and this massive amount of TSH which is being produced overdrives the thyroid gland and results in that when massive amount of TSH work on follicular cells, that forces the follicular cell to produce more T3 and T4. So in these patients, TSH primary problem is with the production of TSH. The TSH is high, and that leads to elevation of T3 T4 level in the blood, and this elevated T3 T4 cannot suppress TSH as this is adenoma growth, but it can suppress TRH. So in these patients, what really happens, primary problem is in elevated TSH, and elevated TSH lead to elevated T4 level, right? But T4 level, once they are elevated, they cannot suppress the TSH. In this particular case, we call it pituitary hyperthyroidism, but it can suppress TRH. Now we come to some very rare cases. That sometimes they're very rare cases, and there is pathology present in disease present in hypothalamus. And in this particular very rare case, hypothalamus is producing excessive amount of unduly excessive amount of TRH. If TRH is very much excessive, it will drive the anterior pituitary to produce excessive amount of TSH, and this excessive amount of TSH will force the thyroid gland to produce excessive amount of T3 T4. But this excessive amount of T4 cannot suppress TSH and TRH. So with these patients, real problem is in excessive production of TRH, which lead to excessive production of TSH, and which eventually result into excessive production of T4, and which leads to thyrotoxic status. Now the basic principles in the management of hyperthyroid state is, for example, if you have a patient with Graves' disease, number one is you give the patient propranolol because it blocks the beta adrenergic receptors, right? And beta adrenergic clinical features, which those clinical features in the patient which are due to overstimulation of adrenergic system, right? Those clinical features will subside, like anxiety will be less, like tremors will be less, tachycardia will be less. Secondly, you give some drug which should reduce the production of T3 T4. For example, you can try thiopropyl, uracil or carbimazole. Already we have discussed in previous lecture that this can inhibit the thyroid peroxidase enzyme and reduce the synthesis and release of T3 T4. Number 3 is that you can go for, if these measures are not working well, you can think of surgery. And when you think of surgery, usually we go for subtotal thyroidectomy. Subtotal thyroidectomy, and you have to specially take care for two things. Number one, subtotal thyroidectomy means you remove a part of the thyroid gland, right? And when you are doing total thyroidectomy or subtotal thyroidectomy, you have to take special care not to remove the parathyroid gland along with the pathological tissue. And number two, recurrent laryngeal nerve is passing upward behind the thyroid gland, that should not be injured. Another option which is there is that you administer patient with radioactive iodine. And if you give radioactive iodine orally, this radioactive iodine is from GIT absorbed, go to the circulation and concentrate into thyroid gland and gradually destroy the thyroid gland, so hyperfunction is finished. But one of the long-term complication is it will produce hypothyroidism. Now we are going to talk about hypothyroidism. First of all, we have to define what is hypothyroidism. Hypothyroidism is a clinico-pathological syndrome which results due to chronic deficiency of action of T3 and T4 on the tissues, right? Now, causes of hypothyroidism are very different types. There are many causes of hypothyroidism. The detail we'll discuss in pathology. Here we'll discuss hypothyroidism in short, right? Number one, causes of hypothyroidism may be related with the thyroid gland, right? Most of the patients who have hypothyroidism, they have normal hypothalamus, they have normal anterior pituitary, but in spite of normal hypothalamus and normal anterior pituitary, thyroid gland cannot produce enough T3 and T4 which is required by the body. This type of hypothyroidism is called primary hypothyroidism. Now let's go into detail of primary hypothyroidism. What could be the reason why thyroid tissue cannot produce enough T3, T4? What's wrong with this? One reason may be autoimmune disease, that there is some autoimmune disease which destroys the thyroid tissue. It's quite possible that there is immune headquarters, and from the immune headquarters autoantibodies are produced, and these autoantibodies attack the thyroid tissue. Now, when these autoantibodies attack the thyroid tissue, they destroy the follicular cells. And if there is progressive destruction of follicular cells, eventually what happens? The capability of thyroid gland to produce T3 and T4 is reduced, right? And this type of autoimmune disease, which destroys the thyroid gland and eventually produces hypothyroidism, it is called Hashimoto's thyroiditis. It is called Hashimoto's thyroiditis. Now, something very important to understand. You must be able to differentiate Hashimoto's disease from Graves' disease. You know, Graves' disease is also an autoimmune disease. Hashimoto's thyroiditis is also an autoimmune disease. But in Graves' disease, autoantibodies which are directed against the follicular cells, they actually stimulate TSH receptors and they lead to overproduction of T3 T4. So what we can say, the autoimmune processes in Graves' disease are basically stimulatory for thyroid gland. Opposite to that, in Hashimoto's disease, autoantibodies which are produced, they do not stimulate the thyroid tissue, rather they destroy the thyroid tissue. And because they destroy the thyroid tissue, eventually there is hypothyroidism. So again, in Graves' disease, patient has autoimmune reaction. In Hashimoto's, patient also has autoimmune reaction. But in Graves' disease, autoimmune reaction is stimulatory to thyroid tissue. In Hashimoto's disease, autoimmune process is destructive to it. So one cause was autoimmune thyroiditis, which eventually leads to hypothyroidism. Another cause may be, fine, iodine deficiency. In some geographical areas, the iodine is not present in enough amount in the soil and in the plants and other dietary elements. Remember, we don't need much iodine. For full year, you need only 50 milligrams of iodine. For 1 year, how much iodine you need? Just 50 milligram per year. Iodine is needed by everyone to produce enough amount of T3 T4. It means weekly you need how much iodine? Yes. Can you calculate, madam? Weekly. Do you know how many weeks are there in 1 year? Yeah. Do you know how many weeks are there in 1 year? 40. Oh, you missed 4 weeks. Usually I think there are 52 weeks. Maybe I'm wrong. Anyway, if we talk about weekly, right, there are 52 weeks I think in 1 year, right? And if you talk about weekly, it means approximately you just need 1 milligram iodine per week. It's a very small amount. Unfortunately, in some areas in the world, even this small amount cannot be provided by the diet. Now, which are those areas? Especially the areas, iodine deficiency areas are those geographical areas which consist of foothills of big mountains. Foothills of big mountains. From thousands of the years, water is coming from big mountains, and with rapid speed it is washing the iodine from the foothills, and then water lay down all the iodine usually in the plains. Is that right? So usually the people who live on the foothills of big mountainous areas, those people get chronically low iodine in the diet. So what happens? Generally that population become iodine deficient. Of course, when you are iodine deficient, the production of T3 T4 is reduced or increased? Reduced. And when production of T3 T4 is reduced, what really happens? The TSH will increase. TSH will overstimulate the thyroid gland, and thyroid gland will produce more and more thyroglobulin, but it cannot iodinate that thyroglobulin properly. So what really happens? That in those patients who are living in iodine deficient area, right, and if their food is not fortified by the iodine or salt is not iodized properly, then what really happens? That due to chronic deficiency of iodine, their production of T3 T4 is less. Due to that reason, there is less negative feedback for TSH and TRH. So TRH level and TSH level become high. And when in the blood TSH levels are high, these high levels of TSH overstimulate the thyroid tissue, and thyroid cells, follicular cells, keep on producing a massive amount of thyroglobulin, but that thyroglobulin is not getting iodinized. Result is that thyroid gland will become larger than normal. So when thyroid becomes larger than normal, we call it goiter. And because this type of goiter is very common in those geographical areas where iodine deficiency is there, we call such type of goiter endemic goiters. What do we call it? Endemic goiter. What are endemic goiters? Endemic goiters are seen, endemic goiters which are due to iodine deficiency. They are seen in certain geographical areas in the world, right? And where many people in the population have goiter, right? In these patients, T3 T4 levels, free T3 T4 levels are chronically low. Of course, total T3 T4 are also low. And in these patients, of course, TSH levels are high. The same way TRH levels are also high. Then another cause of hypothyroidism is use of goitrogenic substances which interfere with the synthesis of thyroid gland. Can you tell me some substances which are goitrogenic? Ladies usually know such substances, but they don't want to tell. Is that true? You know that flour, cauliflower, cabbage, turnips. You know what is turnip? T-U-R-N-I-P. It's a vegetable these days. So turnips and cabbages, if for example you say for full 6 months you will only live on turnips and cabbage, maybe due to some psychotic reasons, right? What will happen to you? Many problems will occur, including hypothyroidism, because turnips and cabbages are having certain substances which interfere with the thyroid gland to produce enough T3 T4. Then there may be hormonodysgenesis. Hormonodysgenesis means that in some people the enzymes and transport mechanisms of certain proteins which are part of follicular cells and which play a role in T3 T4 formation, storage and release, those proteins are defective. For example, in some people, iodide trapping pumps are not functional properly. In some other people, maybe thyroid peroxidase is not functioning well. In some other people, maybe deiodinase enzyme is not working well. So if you have such iodide transporting problem or enzymes problem, it means that in spite of normal iodine supply, person is unable to produce enough amount of T3 and T4. And if you are unable to produce enough amount of T3 and T4, you will become chronically hypothyroid, and eventually that hypothyroidism will lead to what? Yes, high TSH and TRH, right? So today onward, whenever you see a patient who has come with the clinical syndrome of hypothyroidism, you have to do his which test? Which test you will love to do? T3 T4, again remember, free level, but most sensitive test is TSH. Actually these people have a low free T3 or low free T4, and along with that there must be elevated TSH. Because if someone has low T3T4, with that he has low TSH, it means problem is in the pituitary, not in the thyroid. So people have a primary problem in the thyroid, they must have low T3T4 free, along with that they must have elevated TSH. Is that right? Am I clear? Now, other causes for hypothyroidism may be fine. You may have a patient with hyperthyroidism or Graves' disease. You have treated him with radioactive iodine, and radioactive iodine has concentrated into his thyroid gland and gradually destroyed the thyroid gland. So such patient of Graves' disease from hyperthyroid state will become euthyroid, and eventually it may become hypothyroid. Is that clear? Right? Now, these were just a few causes of primary hypothyroidism, right? Remember, in primary hypothyroidism, you have to give enough T4 so that TSH should be suppressed and its level should become within normal range. Let me repeat it again. That actually, these patients who are suffering with primary hypothyroidism, usually we treat them with T4, thyroxine, exogenous thyroxine. And exogenous thyroxine, when we are given, this exogenous thyroxine should be given in sufficient amount, not only to alleviate the clinical features of hypothyroidism, but biochemically you must make it sure that TSH is suppressed enough that after the treatment of T4, TSH level is within normal range. Is that clear? Right? And again, remember one thing. If today you get a patient with hypothyroid state and today you start on the thyroxine tablets, right, it will take few weeks that gradually person's basal metabolic rate will go up and patient's clinical features will disappear related with the hypothyroidism, right? Having said all of this about the primary hypothyroidism, let's talk about less common cause of hypothyroidism, that is pituitary hypothyroidism. In pituitary hypothyroidism, the real problem is in the pituitary gland. That in the pituitary gland, those cells which are thyrotrophs cells are those cells in the pituitary which are supposed to produce TSH. If thyrotrophs are destroyed in the anterior pituitary, or they are not producing enough TSH, or there is global destruction of anterior pituitary, right, due to some vascular event, right? There are some conditions like we call it Sheehan syndrome. In Sheehan syndrome, what happened that postpartum, when a woman after the delivery she has a heavy bleeding, right? She goes into shock. Sometimes the shock produces spasticity and thrombosis in the vessels perfusing the anterior pituitary, and in such women, if anterior pituitary undergoes infarction, right, the condition is called Sheehan syndrome. In Sheehan syndrome, what multiple problems occur. One problem is TSH is chronically low, and then what will happen that thyroid follicular cell will be no more driven by the TSH. Eventually patient will become T3 T4 deficient, and this lady will develop features of hypothyroidism. Is that right? Moreover, such patient will also develop deficiency of due to ACTH and cortisol deficiency, and deficiency of FSH, LH, and you can say fertility functions are also, reproductive functions are also disturbed. Anyway, again let me repeat it. Whenever problem is, the basic problem is in anterior pituitary, and anterior pituitary is unable to produce enough TSH, right? And if TSH circulating level is chronically low, eventually T3 to T4 level production by the thyroid gland also become low, right? But TRH level will go high. Is that clear? So in these patients will have low T4, but primary problem is low TSH. So low TSH is due to primary disease in the pituitary, and low TSH is unable to produce enough T4 and unable to suppress the TRH. So what we can say, TSH will go down, resultantly T4 will also go down, but TRH will remain high, right? Then very rare type of hypothyroidism. A very rare type of hypothyroidism is a condition in which basic problem is in the hypothalamus, and there is something wrong with the hypothalamus, and hypothalamus is unable to produce enough TRH, right? Or pituitary stalk is disturbed. Whatever the cause, if TRH is in low amount reaching to anterior pituitary, either TRH is not produced or pituitary stalk is damaged, right? If chronically TRH is low, then TSH will be also low, and it means thyroid gland will be underdriven. Result will be chronically T3 T4 will be low, and patient will develop clinical features related with hypothyroidism. In this particular case, TRH will be low, which is a real disease, real problem that will result into low TSH, and low TSH will eventually result into low T4. Is that clear? So we have briefly discussed causes of hypothyroidism, and now we come to the clinical features of hypothyroidism. The good news is that clinical features of hypothyroidism we have already discussed in the last section when we were talking about the clinical features of hyperthyroidism. At that very time, we compared the clinical features of hypothyroidism and hyperthyroidism. Is that right? So now we'll go ahead and we'll talk about a very special type of hypothyroidism, which is called cretinism. Now we are going to talk about a very special type of hypothyroidism, which is called cretinism. This is a hypothyroidism which is present from the birth, right? It's a hypothyroidism in babies, right? Basically, it can be of two types. There can be congenital hypothyroidism, congenital cretinism, or there can be endemic cretinism, right? Now, in congenital cretinism, what really happens that, let's suppose a baby is born, but baby does not have this baby does not have a thyroid gland at all, or thyroid gland is there, but it is having abnormal enzymes and production of T3 T4 is dangerously low, extremely low. Is that right? Such baby, when they are born, initially they are normal. The reason being that T4 can cross the placenta, and during the intrauterine life, fetus has been getting at least some amount of T3 T4 from the mother, right? But after the birth, after a few weeks of the birth, baby become gradually sluggish and slow and develop hypotonia and loses the, you can say, even baby does not have enough power to suckle the milk. Now, congenital cretinism is when baby is born with the problem. And endemic cretinism is a condition in which an area is extremely deficient in iodine, right? There's severe deficiency of iodine in the soil and in the dietary products, right? And child develop severe deficiency of iodine from the very beginning of the life. As it grows, it suffers more and more features of hypothyroidism. Now, the special features of these cretin babies are that, number one, they have short height, they're dwarfs because skeletal growth is not well. Number two, they are obese. Now, why they are obese? The reason being that even though deficiency of T3 T4 affects the skeletal growth as well as soft tissue growth, but skeletal growth is affected more and soft tissue growth is affected less. Due to that reason, height remains very small, skeleton remains very small, but soft tissue as in comparison to the skeleton is slightly more. Sometimes even tongue becomes so large that it becomes very difficult for the baby to hold it in his mouth, right? It may be protruding all the time. With that, another very important problem is that this baby has mental retardation, and this is something you should not forget. This is extremely important point, because this is one of the commonest cause of mental retardation which can be prevented, right? Why these babies develop mental retardation? Because when there is chronic deficiency of T3 T4, synapses don't develop well, dendrites out of the neurons don't grow well, and myelination of neuron is not well. Due to that reason, when cerebral cortex is not developed well, there is severe mental retardation. And if later on you replace T3 T4, especially thyroxine can be given exogenously, unfortunately mental retardation cannot be reversed properly, right? Second problem is the basal ganglia are even affected. When basal ganglia are not developed properly, so babies develop some clinical features related with the extrapyramidal system like rigidity. Some of these babies develop rigidity, right? And third important problem which is there is that in the absence of T3 T4, your cochlear system, inner ear development, cochlear system does not develop well. And if your cochlear system is not developing well, it means baby cannot hear anything. Of course, baby cannot talk well, and we call this condition deaf mutism. We call this condition deaf mutism. Remaining clinical features are same as in adult. For example, total BMR is less, hair are thick and unmanageable, skin is thick due to collection of chondroitin sulfate and proteoglycans under the skin, right? Heart rate is low, so blood pressure is low. In these patients, cholesterol is not catabolized properly, so they have hypercholesterolemia, right? There are slow reflexes. So all other features are same as in adult hypothyroid. The major difference is if a person has hypothyroidism from the childhood, he will have mental retardation. But if you are an adult and initially you have had enough T3 T4 and your mental development and maturation has been normal, and you are in the age of, let's suppose, 35, and then you develop T3 T4 deficiency, right? If you develop T3 T4 deficiency at that very time, you will develop psychomotor slowing, but you will not develop mental retardation. Again, let me repeat it. Mental retardation develops when T3 T4 deficiency is very severe from the very beginning of the life. Mental retardation. And due to this reason, because this is preventable cause, right? So due to this reason, what we do in America or in other many Western countries, every child which is born undergoes screening for hypothyroidism. So that even though hypothyroidism in a newborn is not a common thing, but still it's very important to diagnose, because you just give exogenous thyroxine and you prevent the whole cretinism. Is that right? So most important point which you have to remember out of this is that cretin babies which are due to hypothyroidism, they are short, they are obese, they are stocky, and they are mentally retarded, because the neurons don't develop well, they don't grow well, myelination is not well, dendrites don't grow well, right? And along with all these things, right, the mental retardation is not reversible completely if you introduce T3 T4 at a later stage. Due to that reason, it's very much important that every baby which is born should be screened for the hypothyroidism, so that if there is real deficiency of thyroid hormone, it should be replaced as early as possible.